Good morning, good afternoon, or good evening. This is Lisa Whitehead, and today we are studying Chapter 28, the Reproductive Systems, in Bio 205 at Illinois Central College. Learning outcomes for today's lecture include the following: to identify the principal components of the human reproductive system and summarize their functions, compare the production of gametes between a male and a female in regards to quantity produced and the timing of gamete production in their lifespan, identify the structure and function of the male reproductive system organs and accessory glands in the production, maturation, storage, and transport of semen. List the pathway of spermatozoa from production to ejaculation, and in this, a subsection being to define capacitation and determine where it occurs. Explain why the testes are located in the scrotum outside of the body, and to review mitosis and meiosis, and outline the processes of meiosis and spermatogenesis. Also, to summarize the hormonal mechanisms that regulate male reproduction, to describe sperm anatomy and identify organelles it has and those that are missing, identify the components of the female reproductive system, compare the myometrium and the endometrium in the uterine wall, outline the processes of meiosis and oogenesis, understand the functions of the female reproductive organs in oogenesis, identify the phases and events of the ovarian and uterine cycles, and lastly, to summarize the anatomical, physiological, and hormonal aspects of the female reproductive system. The reproductive system is literally the only system that is not essential to our lives. We could lose our ability to procreate and still carry on otherwise completely normally. We can still breathe air, our cardiovascular system still functions, our neurological system still functions, but it is not really affected by the reproductive system. That's not to say that it doesn't affect other systems. Our reproductive system affects our entire body in myriad ways. And so the male and female reproductive systems are both very different, but the overall goal is the same. It is to produce and store specialized reproductive cells that can be combined to form new individuals. Our reproductive organs are also responsible for secreting hormones, and those hormones are going to play a major role in maintaining normal sexual function and reproductive ability in humans. Today's lecture follows the outline of our textbook, so that way you can follow along with your studying. So first, we're going to discuss the production of gametes, and then we're going to move into discussion of male anatomic structures of the reproductive system. And then we're going to look at the process of spermatogenesis and hormones that control the male reproductive system. Next, we'll move into female anatomic structures, and then oogenesis and the female hormones of control. Then we'll move into the physiology of sexual intercourse, finally, development and aging, and then interactions between different systems. So let's get started. The reproductive system exists essentially to continue existence of the species. And so there are a number of different structures that are a part of our reproductive system that all have the same end, but they play a different role, like different athletes on a field playing football. So first off, we have gonads. And the gonads are going to be our reproductive organs that produce gametes. And gametes are the reproductive cells that are responsible for meeting and replicating in order to create a new individual. The gonads also are responsible for producing hormones. So both the ovaries and the testes produce hormones that have far-reaching impacts throughout our body as well as on our actual reproductive processes. We also have ducts that are a component of our reproductive structures. And these ducts are there to both receive the gametes and then to transport them from where they're produced to a point at which they can meet the opposing other gamete and therefore engage in reproduction. We also have accessory glands, and these accessory glands are there to secrete fluid into those ducts, which helps to transport the gametes and nourish them on their way. We also have perineal structures, and these are altogether known as our external genitalia. And this is what we would think of when you look at genitalia on the outside of a body. These are the perineal structures we are referring to. The reproductive tract is essentially all of the chambers that we have and all of the passageways that are going to connect our exterior body to the reproductive system. So it has ducts and a number of other passageways as well included in this. The male and female reproductive systems are functionally quite different. However, together, they work together to be able to produce a new individual. So the female reproductive system will produce, on average, one gamete per month. And this is an ovum, which is singular for egg. In plural, it's ova. The female is also responsible for retaining and nurturing the zygote as the zygote becomes an embryo and eventually a fetus and then is delivered and is an infant. The male reproductive system, on the other hand, has a different approach. They produce millions, like billions even, 500 million of sperm per day. So it's quite a great number of gametes. And there is no responsibility in the male reproductive system for retaining or nurturing a zygote. The male reproductive system is responsible for producing male gametes, which are spermatozoa, as well as for producing male sex hormones, which are known as androgens collectively. And these are both produced in the male gonads, known as the testes. Please note that testes is plural, and testis, spelled I-S instead of E-S, that is the singular. So if you refer to the right testis, it's I-S, but if you're referring to the right and left testes together, it's T-E-S, T-E-S. In the female reproductive system, there are ovaries, which are the female gonads, and the ovaries are responsible for releasing an immature gamete known as an oocyte each month. Sometimes there will be 2 released each month, and in that case, if both were fertilized, we could result in fraternal twins. Also, by the way, it's possible for a single oocyte to be fertilized and for it to split, in which case then you would have identical twins. So once that gamete is released, it will be maintained by hormones that are also produced by the ovaries. But the ovaries produce hormones that don't just maintain a gamete or an ovary. They have far-reaching effects throughout the entire female body. The uterine tubes are responsible for carrying that oocyte from the ovary to the uterus. And if the sperm is able to reach the oocyte, then we'll have fertilization. And at that point, the oocyte matures into a full ovum. The uterus is going to be there as the organ that encloses and supports the developing embryo from implantation all the way through labor and delivery. And the vagina is going to be the passageway that connects the uterus with the exterior of the body. Now let's take a little bit closer look into the structures of the male reproductive system, including the testes, the duct system, accessory glands, and the penis. When a sperm is being developed, it will originate in the testis on either the right or the left side, and as it matures, it will mature in the epididymis, and then travel through the ductus deferens, meet the ejaculatory duct, and finally pass through the urethra before it is expelled. Now, there are accessory glands that are going to secrete fluids into this duct system to support the sperm as it goes through the whole entire process of maturing and also so that way it can be expelled. So there are seminal glands, the prostate gland, and also the bulbourethral glands, which all play an important role in helping sperm through the passage. Just so that way I'm sure that you don't confuse the testes and the scrotum, because they are 2 totally separate things. The scrotum is actually the skin-like sac that encloses the testes, whereas the testes are the reproductive organs themselves. So the testes are about 5 centimeters long, 3 centimeters wide, and about the same in terms of thickness. Each testis is going to weigh 10 to 15 grams, and it hangs in the scrotum, and it can move around inside the scrotum itself. The scrotum is just a support structure that hangs inferior to the perineum and anterior to the anus, while also being posterior to the base of the penis. On cross-section here in figure 28.1, you can look at the major structures of the male reproductive system. So looking at the image at the right, you're going to see at the top, the largest structure that you see is the urinary bladder, and what it rests upon is a structure called the prostate gland. And so the prostate gland is there, and as you can see, you have 2 different structures that are moving along through, and it bifurcates right at the base of the prostate. And this is our ejaculatory duct coming from the seminal vesicles, and as they both meet up, the right and the left ejaculatory ducts, you will find that they come together into the urethra. And at the top, this is considered the prostatic urethra while it is still in the prostate gland. And as it extends down into the penis, it becomes the penile urethra. Either way, it is connected, and that is the passageway for urine that comes down through the bladder. But also, this same passage is important because the seminal vesicles, which are up sort of posterior and superior to the prostate gland, those are responsible for providing some fluid that will support sperm as they travel through their passage. So the spermatic passage is very different. It's going to start off at the testis, which is down at the very bottom of the screen here, as that egg-shaped structure in the scrotum. And so once it's produced in the testis, it will then travel up through the epididymis, mature there, and then through the epididymis you can see that there's kind of like a curve. It's almost like you've got something hugging the outer surface of the testis, and then it bends down, and then the extension of it curves all the way up by the urinary bladder and loops around, and it's hard to see in this image what's going on. But either way, the point is the same, that the testis is where the spermatozoa originate, they mature in the epididymis, they travel up through the ductus deferens, which is also known as the vas deferens in some textbooks, and then it's going to come back around and be able to meet up with products from the seminal vesicles, because it deposits right into the prostate gland where the seminal vesicles are via its own duct. So it's in the prostate gland where the sperm will meet up with fluid from the seminal vesicles, and then as the prostate gland contracts, that will help to propel the sperm down through first the prostatic urethra, then into the penile urethra, and then out. The blood supply and innervation that is for the testis is supplied via the spermatic cord. And the spermatic cord, as you can see in figure 28.1, is not just a single strand, but rather it's an entire tubular type structure that includes layers of fascia and muscle, and it stretches from the abdominal pelvic cavity to the testis, and it wraps around the ductus deferens, blood vessels, nerves, and also the lymphatic vessels that support the testes. So each of these begins at the entrance to the inguinal canal, and the passageway goes right through abdominal musculature. So this will drop down into the scrotum, so that way there's a constant blood supply and support for the testis. Within the spermatic cord, there are several vascular structures, and this includes the deferential artery, the testicular artery, and then we have the pampiniform plexus of the testicular vein in there as well. And as you can see, that plexus is rather complicated looking. There are also nerves that support the testis, and these come from branches of the genitofemoral nerve that arise from the lumbar plexus. An interesting clinical correlation of this spermatic cord is that inguinal hernias are incredibly common here. And this is because there is a weak point in the abdominal wall at the specific site. And so what a hernia is essentially just a protrusion of visceral tissues into a place where they're not supposed to be. So this image on the right is from the Cleveland Clinic, and it's showing you where an inguinal hernia is versus a femoral hernia versus an umbilical hernia. And it's all the same thing. We just have abdominal tissue that pokes through a weakness in a soft spot and allows for sometimes even a full loop of small intestine to fall into that location. And once it's there, it can torse or twist. And upon twisting, then it can lose its vascular supply, and then it becomes necrotic and dies. And as you can imagine, that would have terrible repercussions for our bodies, because that means that the gastrointestinal contents that are present inside the small bowel would potentially rupture, and then eventually that would be throughout our peritoneal cavity, which would result in a massive reaction from our body. Never mind that. Also, not having any food processed through your gastrointestinal tract because of a stoppage, because of a hernia that has obstructed passage, will result in all sorts of discomfort, in addition to vomiting and potentially a number of other side effects. The scrotum is going to be divided into two separate chambers, the right and the left, by the raphe of the scrotum. And the raphe is a raised thickening in the scrotal surface. Each testis is going to be present in its own separate scrotal cavity. And then the tunica vaginalis is going to be a serous membrane that lines the entire scrotal cavity, and it's there specifically for the purpose of reducing friction between the opposing surfaces. So keep in mind that the testis within the scrotum can move, and so that tunica vaginalis has an important role in allowing that movement to occur freely. So there are two layers of the tunica vaginalis. There's the outer and the inner layer. The outer layer, just like other parts of our body, is known as the parietal layer because it is peripheral, and the visceral layer is right upon the viscera or the inner organs, so that is the inner layer. There are two primary muscles that exist within the scrotum that are important for scrotal maintenance of temperature and as well as for reproductive purposes. The first is the dartos muscle. The second is the cremaster. So the dartos muscle is composed of smooth muscle, and what it does is it allows for the testis to become slightly elevated and also wrinkles the scrotal surface. The cremaster, however, is composed of skeletal muscle, and it's important for being able to contract, which will allow for the testis to be pulled in closer to the body if heat is needed, or if the body is very warm. It allows for relaxation to be able to drop the testis further away from the body to allow for greater cooling. There's a reflex called the cremaster reflex, and what it does is it moves the testes closer to the body based upon stroking of the skin on the upper thigh. The testes require a temperature that is 1.1 degrees Celsius lower than body temperature in order to develop sperm that develop normally. So in order to achieve that, we have to have movement of the testis either closer to the core body or further away from the body in order to achieve that 1.1 Celsius degree difference. So the muscles that we have, including the cremaster and the dartos, are there to either relax or contract, to move the testis away from the body, to cool down, or towards the body if cold. And it is specifically these muscles, specifically and mostly the cremaster, which is responsible for maintaining temperatures of the testes that are acceptable and amenable to sperm development. Now let's look at some of the anatomy of the testis. So we've already talked about the tunica vaginalis, which is the outer aspect of the scrotum. But there is another inner layer, and that is called the tunica albuginea. And the tunica albuginea has the word alba in the very beginning, which means white. If any of you are gardeners, you'll know that alba varieties of any plant will be the white variety of the flower. So in this case, the tunica albuginea is white and smooth, and this is deep to the tunica vaginalis, and it's composed of a dense layer of connective tissue that's composed primarily of collagen fibers. This is continuous with fibers that surround the epididymis, and the fibers from this will form the septa of the testis that converges upon the entrance to the epididymis. The tunica albuginea is also responsible for supporting blood and lymphatic vessels of the testis and also of the efferent ductules. Figure 28.3 shows you the histology of testes, and this is what a testis looks like microscopically at 26 power. So first we have septa that will subdivide each testis into lobules, and then those lobules are going to contain about 800 very narrow, delicate, and very tightly coiled seminiferous tubules, which are the site of sperm production. In the laboratory, because I've worked as a PA, I would receive occasionally an orchiectomy specimen, which is a testicular resection of just the testis itself. And this can be for a number of reasons, ranging from malignancy through trauma to a number of different things. But either way, one of the things that we do in the laboratory to determine if the tissue is structurally intact is we test its integrity by taking a pair of forceps and grabbing these seminiferous tubules, which are about 1 millimeter in diameter, and we just grab on it and pull them up to see if we can string it out with ease as a measure of its integrity. So each of these really tightly coiled seminiferous tubules is going to be about 80 centimeters long, which is almost as long as a yardstick. And that's nearly 1/2 of a mile of seminiferous tubules end to end in each testis. So these will connect to the rete testis, which is a network of interconnected straight tubules. And the efferent ductules are going to connect the rete testis to the epididymis. Around the seminiferous tubules, there's a connective tissue capsule, and there's areolar tissue that's going to fill the spaces in between those tubules. So within those spaces, there's going to be two things: blood vessels and large interstitial endocrine cells called Leydig cells. And the Leydig cells are important for producing androgens, which is the dominant male sex hormone. So testosterone is primarily the #1 androgen that's being produced in the Leydig cells. Figure 28.4 shows you a cross-section through seminiferous tubules on the left, and then on the right, it's zoomed in to a power of 350 to show you a cross-section through a single seminiferous tubule. On the left, you'll notice that there are multiple tubules, and you'll see that they're labeled that they have both late spermatids and early spermatids, as well as actual, normal, mature sperm. All of these are present here because this is where they originate. So if we zoom in on the right and look at image B, you're going to see a number of different types of cells there. There are the interstitial endocrine cells responsible for producing androgens, specifically testosterone. These are our late egg cells. We also have dividing spermatocytes that are in the process of becoming spermatids, moving into the lumen, which is the opening in the center of the tube. There are also heads of maturing sperm that you can see down at the bottom, indicated by the lowest pointer. And there are also nurse cells present, known as Sertoli cells, which we'll discuss very shortly. The testes are responsible for producing sperm, but these sperm are still immobile, meaning they can't actually move yet. So if they can't move yet, they're not capable of fertilizing an oocyte. This is for two reasons. The first is they can't physically get to where the oocyte is if they're able to move. The second reason is even if they were placed right immediately adjacent to an oocyte in a petri dish, if they're immobile, they won't be able to break through the barrier of the oocyte wall, and therefore they would not be able to fertilize the oocyte. So, therefore, we have other parts of our reproductive system that chime in and help support the sperm so that way they can actually fertilize an oocyte. So there are other parts of our reproductive system that help them mature functionally and nurse the sperm on their way, hold them in storage until they're ready, and then transport them at the appropriate time. The immobile sperm that come from the testes are going to be moved by cilia, which will help to wave along in a pattern along those efferent ductules into the epididymis. The epididymis is the start of the male reproductive tract, but it is not the start of the male reproductive system. Sperm cells will originate in the testis, but it is the tract that carries them from one point to the other. So this is the beginning of them being carried from the testis to the exterior of our body. So the epididymis is a tube that's coiled, and it's about 7 meters long in total, and it's bound to the posterior border of each of the testes. So there's a head, a body, and a tail on each epididymis. So let's look at the image here on the right. You'll notice up at the very top of the image in the center, you'll see that is the head of the epididymis, and then the body kind of wraps down posteriorly along the testis down to the very bottom where it is then called the tail. And then you'll see as it wraps around and stretches out, it is now the ductus deferens, and the ductus deferens will continue on. So let's take another look at figure 28.5a. You'll see the head of the epididymis is the largest part of it, and this is where it receives sperm from the efferent ductules. The body of the epididymis is on the posterior aspect of the testis, and it wraps around, hugging that posterior aspect. The tail is going to begin at the inferior border of the testis, and then it's going to ascend to connect with the ductus deferens, and this is the primary storage location of sperm. The epididymis is responsible for continually monitoring and adjusting the composition of the fluid that the seminiferous tubules create. It also is there to basically recycle damaged sperm. So any cellular debris or sperm that are damaged will be absorbed in the epididymis. The products of this breakdown are going to be released into the interstitial fluid around the tissue, so it can be picked up by the surrounding blood vessels. And then lastly, it also functions as a site of storage as well as for protection to help facilitate sperm as they mature. The ductus deferens, which has been previously known as the vas deferens and is plurally known as the ductus deferentia, is about 40 to 45 centimeters long and is part of the spermatic cord. So it starts at the tail of the epididymis and it extends, ascending up through the inguinal canal. It curves slightly inferior along the bottom of the urinary bladder and then bends towards the prostate glands and meets up at the prostate at the same site where the seminal glands deposit their product into the prostate gland. So the lumen of the ductus deferens is going to open up broadly into the ampulla of the ductus deferens, and that is at the prostate gland. The wall of the ductus deferens is thick, smooth muscle. The lining of the ductus deferens is with a pseudostratified ciliated columnar epithelium. Flashback to your week 4 of this class. And it is here where there are peristaltic contractions that will propel the sperm as well as the fluid that it's in out. So sperm can live here in the ductus deferens for up to several months, and they're in an inactive state at this point. So at this point, they have very low metabolic rates, and they're just kind of hanging out, almost like hibernating, if you want to think of it that way, waiting for the next step in the process. The male urethra is used by both the urinary and reproductive systems, although in the female reproductive system, the urinary bladder and the urethra is not a part of it. So the male urethra will extend 18 to 20 centimeters from the urinary bladder to the tip of the penis, and we break the urethra down into 3 regions from proximal to distal. There's the prostatic, which is actually inside of the prostate gland. There's the membranous, and that is in between the prostate gland and the penis. And then we have the spongy urethra, which is located within the penis. In the male reproductive system, there are accessory glands that are there to produce fluid that is a component of semen, and this supports the sperm. So ultimately, semen is a mixture of multiple secretions that each come from these glands and has its own distinctive biochemical characteristics. The 3 important glands of the reproductive system in the male are the seminal glands, also known as the seminal vesicles, the prostate gland, and the bulbourethral glands. So the seminal vesicles or seminal glands are bilateral. There is right and left. And you will see that in this image, we are looking at the posterior view of the bladder, and this is actually somewhat postero-inferior. So we're looking kind of up at the bladder anteriorly. And you can see here that we have at the very top of this image, the urinary bladder, and then there are 2 major structures extending out, the ductus deferens, which comes into the ampulla and opens up its product into the prostatic urethra. Then they're also moving just a little bit more inferiorly. We have the 2 seminal glands or seminal vesicles, which also produce their product and secrete it into the prostatic urethra. And then there's the prostate gland, which occupies the majority of the inferior aspect of the image. And then at the bottom, we have extending into the membranous urethra and the penile urethra, we have the bulbourethral glands. The major functions of the accessory glands are to help activate sperm and to provide them with nutrients that the sperm need for motility, which is movement. This fluid is also going to help to propel sperm and the fluids along the reproductive tract, and this is mainly by peristaltic contractions of the smooth muscle. These accessory glands also produce buffers, and the buffers are there to counteract acidity of the environment that they're in. So you can imagine that in the urethra, that urine could be somewhat acidic. So there are buffers within these fluids to help counteract that acidity, so that way residual acidity doesn't damage the sperm. The seminal glands are the first in the line of production among the accessory glands. So seminal glands, also known as seminal vesicles, these are responsible at the very beginning for producing the majority of seminal volume. So they're extremely active glands, and they're only about 15 centimeters long, but they're kind of wide. They're about 5 centimeters by 2.5 cm overall, folded into this compact unit of tubular glands. Earlier, we discussed how sperm are formed in the testis. They will then travel through the epididymis, which is where they mature, and then through the ductus deferens. And that goes up and around. It comes back down to the prostate gland. So this is where everything comes together. The ductus deferens as well as the seminal vesicles are going to deposit their product into the ejaculatory duct, which is just a short little passageway about 2 centimeters long, and this is what penetrates the wall of the prostate. And so this is where the ejaculatory duct is where both the ductus deferens and the seminal vesicles or seminal glands can deposit their product into the prostatic urethra. The prostate is a small but fibromuscular organ that's about 4 centimeters in diameter, and it wraps around the proximal aspect of the urethra right inferior to the urinary bladder. Within the prostate, there are about 30 to 50 compound tubuloalveolar glands, and these are surrounded by smooth muscle fibers. Here's a great clinical correlation. In a normal healthy adult male, the prostate is a normal size, roughly about the size of a golf ball, and as you can see, it rests just inferior to the bladder, so the bladder is releasing its urinary contents through the urethra that starts at the bladder and extends through the tip of the penis. But it has to go through the prostatic urethra first. So as you'll see on the right, the prostate gland here is massive. This is called benign prostatic hyperplasia, and it's incredibly common. And so what ends up happening is with an enlarged prostate gland, you can see here that it's distorting the floor of the bladder. So at the corners of the bladder, there are these areas where urine can puddle. And this is one of the primary symptoms of BPH, which is feeling like you have to go to the bathroom and you have to urinate, although you just went, because there's a little bit of residual urine that doesn't get expelled. And there's a problem with this. Anytime we have stasis, it's like a pond. If you think of a pond without getting aeration and movement, what does it get? Algae. Same sort of idea applies here in the bladder. If the urine just sits still, it's possible for a urinary tract infection to occur at this site. Prostatic fluid is slightly acidic, and this is about 1/4 of the volume of semen, with the remaining components being primarily formed by the seminal glands or seminal vesicles. So this prostatic fluid will be ejected into the prostatic urethra, stimulated by peristalsis that occurs in the prostate wall, which is a fibromuscular wall. Prostatitis is a clinical correlation here, and it is just inflammation of the prostate gland. And this can occur to absolutely any man at all, and it creates a considerable amount of pain. And this is most common in older men. The prostate is a small but fibromuscular organ that's about 4 centimeters in diameter, and it wraps around the proximal aspect of the urethra right inferior to the urinary bladder. Within the prostate, there are about 30 to 50 compound tubuloalveolar glands, and these are surrounded by smooth muscle fibers. Of the 3 glands in the male reproductive system, the bulbourethral glands are the last, and these are the smallest. They're also the most distal. So they're also known as Cowper glands, and these are compound tubular mucus glands that are only up to about 1 centimeter in diameter. These are located right at the base of the penis and secrete a thick mucus that's alkaline. And that alkaline component is really important because it helps to neutralize any residual acid from urine that would remain in the urethra. And an acidic environment could be intolerable to the sperm. So this alkaline mucus is there to help make sure the sperm don't get damaged along the way. So this thick mucus is also there to help lubricate the tip of the penis, and a duct on each gland is going to travel alongside the spongy urethra, and then it empties into the urethral lumen. So as you can see in the image on the right, we have the 2 big round glands, and then they each have their own duct, and that goes right alongside the spongy urethra and dumps into the urethral lumen. Figure 28.6 should probably feel a little bit familiar to you because this is histology that you know from our previous study in Chapter 4. So if you look at this image, what you see are these big mucus glands, and they look very much like our goblet cells, which produce mucin. So these mucus cells are producing that mucus material that is deposited into the spongy urethra by the bulbourethral glands. A typical ejaculation would release 2 to 5 milliliters of ejaculate, and there can be times when a low volume could occur, and if so, an abnormally low volume could indicate problems primarily with the prostate gland or the seminal vesicles/seminal glands, because this is where the majority of the volume is produced. It can also be an indicator potentially of issues with sperm count, although keep in mind that the sperms themselves actually do not take up the majority of the volume of an ejaculate. So sperm count is taken about 36 hours after sexual abstinence has occurred, and the normal values we would expect to see in a normal range is between 20 to 100 million sperm per milliliter of sample. Seminal fluid has the same osmotic concentration as blood plasma, but it is composed of different substances. The 3 things we'll discuss here all have a very specific role to play. The first is that there is a high concentration of fructose in seminal fluid, and this is important because this is an easy, quick source of energy for sperm, so it's easily metabolized by sperm. The second substance in seminal fluid that's different from our blood plasma is prostaglandins. And these prostaglandins play very important roles in both the female and the male reproductive tracts. So in the male reproductive tracts, prostaglandins will help to stimulate smooth muscle contraction to help with the expulsion of seminal fluid at orgasm. But in the male reproductive tract, there's a totally different role. And one is actually very interesting. So you may have heard the old wives' tale that if a woman is full-term pregnant and they wanted to get labor and delivery going, something to do would be to have sexual intercourse. And this is actually a true old wives' tale. It actually works because in seminal fluid, those prostaglandins will stimulate smooth muscle contraction in the female reproductive tract, and this can help to stimulate labor. And then keep in mind that labor is one of our major positive feedback loops. So once it gets started, it goes more and more and more up until the point where you deliver the child. The third thing in seminal fluid is fibrinogen. And fibrinogen is there to help form a temporary clot in the vagina at the site of the cervix. So it allows for the seminal fluid to basically enter the female reproductive tract to seek an egg, but then form a slight clot so that nothing else can get in, including potentially other competitive sperm. The secretions of the seminal glands are slightly alkaline, and this is important because the vagina and also many different parts along the prostatic urethra and the penile urethra will be slightly acidic. So those seminal glands are slightly alkaline to neutralize those acids in both the male and the female reproductive tract, so that way the sperm don't die en route. In semen, there are three main components. The first is sperm, the second is seminal fluid, and the third is enzymes. And these enzymes play important roles. The first enzyme of note is a protease that's there specifically to help dissolve mucus in the vagina. The second is seminal plasmin. And seminal plasmin is an antibiotic protein that's there to help to prevent urinary tract infections, specifically in males. There's a prostatic enzyme that's also present, and this converts fibrinogen to fibrin. So this has the end effect of coagulating semen just a few minutes after ejaculation. And then lastly, there's fibrinolysin, which effectively reverses the effect of that prostatic enzyme because it will liquefy clotted semen after about 50 to 30 minutes. The penis is a tubular organ that belongs to two different organ systems. The first is the urinary system, and the second is the reproductive system. So in the urinary system, the penis acts as a conduit for urine from the bladder through the prostatic urethra, the membranous urethra, and then through the penile urethra. And in the reproductive system, the penis is a conduit of semen. So it passes through the same pathway through the penis, out the external urethral orifice, and that will allow it to come to the outside world or for purposes of reproduction into the female's reproductive tract, specifically into the vagina. The root of the penis is the base of the penis where it's fixed, attached to the body wall, right inferior to the pubic symphysis. The body of the penis is also known as the shaft, and so the body of the penis is the tubular component that is mobile, and the glans penis is the expanded tissue at the head where we have the most distal end of the penis, and this surrounds the external urethral orifice. The dermis of the penis contains a layer of smooth muscle, which is a continuation of the dartos muscle. And remember that the dartos muscle is there that forms the wrinkling of the scrotal sac. There's also underlying areolar tissue, and that areolar tissue, remember, is very loose. And so that's what allows the skin to be able to move freely, both on the penis as well as on the scrotum. And then there's a subcutaneous layer. And in the subcutaneous layer, this is where you're going to find all of the major vessels, the superficial arteries, veins, as well as lymphatic vessels. The technical term for foreskin is prepuce, and this is the fold of skin that surrounds the tip of the penis, attaching to the neck of the penis and extending over the glans penis. So this is mobile skin that can move around, and there are glands inside of it called prepucial glands, and these are in the skin of the neck and the inner surface of foreskin. The prepucial glands secrete a substance called smegma, which is a waxy material, and while it provides some lubrication, it also unfortunately provides an environment in which bacteria can thrive. So as a result, it's very possible and likely that people will get infections in this area over time. Not everyone will, but it is quite common. As a matter of fact, I used to work as a PA at the Medical College of Wisconsin, and there were quite a few foreskin resection specimens that I would receive daily. And this was not from newborns, which is typically done, but rather done for adults because of infection. Circumcision is also often performed at birth, usually within a day or two of birth even. And this is the surgical removal of foreskin. And this is very hotly debated in medical circles as well as in ethical circles, but there are actually some medical indications by which it's helpful. And the reason by which people choose to do this for medical purposes is because there is proven documentation that two things occur. When somebody is circumcised, there is less of that smegma present, and there is nowhere really for it to hide, even for the little bit of it that is there. So what that means is it prevents the risk of infections, and this includes both your regular run-of-the-mill staphylococcus and streptococcus type infections that are omnipresent, but it also helps prevent some sexually transmitted infections. There's also data present that circumcision helps prevent penile cancer. I've also read some data that shows that one of the infections that is reduced as a risk is HIV. So there are medical purposes for circumcision, but it is a very personal decision for people. The majority of the tissue in the penis is composed of erectile tissue within 3 separate cylindrical columns of tissue. And so this erectile tissue is present in the body of the penis, deep to the areolar tissue, which allows for movement, and it's within a dense network of elastic fibers that encircles the internal structures of the penis. So this consists of a network of vascular channels that are separated by these partitions, to some extent, of elastic connective tissue and smooth muscle fibers. So figure 28.7 on the right gives you an overall layout of a cross-sectional view through the penis. So you can see that there are 2 main structures in purple, and these are the corpora cavernosa. And then below that, you'll see there's one circular area that is more in red, and that is the corpus spongiosum. So those are the 3 columns of that erectile tissue present in the penis, composing the majority of the tissue in the penis. At rest, the erectile tissue in the penis will have reduced blood flow, and this is basically accomplished by the arterial branches and the muscular partitions. The arterial branches will be constricted, reducing blood flow into the erectile tissue components, and the muscular partitions are also tense, which will also restrict blood flow into the erectile tissue. However, during erection, things change. At this point, what happens is the penis will stiffen and elevate to an upright position because of blood flow into the erectile tissue. The corpora cavernosa are the 2 cylindrical masses of erectile tissue that compose the majority of the volume of the penis. And in figure 28.7, you can see those areas in purple occupying the majority of the cross-sectional view of the penis. So these are present under the anterior surface of the flaccid penis, which, interestingly, by the way, in anatomic position, would actually be the posterior surface, because in anatomic position, the penis is erect. So be careful to note this. When you're working clinically, if you find a lesion, for example, that you need to describe that might be on the quote-unquote underside of the penis, you would normally think, well, that would be the posterior. But when you're describing it medically in anatomic position, the penis is erect. So the underside of the penis is now the anterior surface. So the corpora cavernosa are separated by a very thin septum, and they're encircled also by a dense collagenous sheath all the way around. These diverge at their bases, which forms the crus of the penis, and each crus is bound to the ramus of the ischium and the pubis, which are major hip bones. You'll cover this in 206 when you get into the skeleton. And they're also connected by these tough connective tissue ligaments that extend to the neck of the penis. So the erectile tissue surrounds a central artery, which you can see in the image here to the right. Both of the corpora cavernosa have a central artery within the very center that is highlighted in red. The corpus spongiosum is that 3rd component of erectile tissue that you see in this image here in red. So this is a more slender column of erectile tissue, and it surrounds the distal portion of the urethra. So it extends from the tip of the penis, and then it expands out to form the glans penis. So this is surrounded by a sheath that has more elastic fibers than that of the corpora cavernosum. And it also has erectile tissue that has a pair of small arteries in it. And this tissue is going to surround another central artery. Figure 28.7b shows you the male reproductive tract in general and all of the structures that are involved. So first, starting up at the top, we have the ureters, and the ureters are conducting urine from the kidneys to the bladder, which is the muscular holding tank for urine until it's time to urinate. So urine, when it's time to urinate, will go through the passage of going through the bladder, through the prostatic urethra, the membranous urethra, and out through the spongy urethra, through the external urethral orifice and out. And then we also have the reproductive system, which is showing you that sperm will be formed in the testis. They go through the head of the epididymis, the body, the tail, and then curve around and into the vas deferens or the ductus deferens, which, as you can see, is curving around and occupies up at the top of the image those two wide tubes that are coming along, and they will deposit into the prostate, right next to the seminal vesicles or seminal glands. So the prostate then is our fibromuscular organ, which can contract at time of orgasm to be able to help propel seminal fluid out. So seminal fluid will then continue to flow through the prostatic urethra and then through the membranous urethra, at which point then we get the addition of fluid and mucus from the bulbourethral glands. It will then flow through the spongy urethra and out through the external urethral orifice as well. Let's look at section 28.3, spermatogenesis and the male reproductive hormones that control our reproductive system. Before we discuss spermatogenesis and the hormones that control the male reproductive system, we're going to discuss mitosis and meiosis just briefly. So the first part we need to discuss is mitosis, which is a part of somatic cell division. And the somatic cells, again, are the cells throughout our body, like our muscle cells, our skin cells, our liver, hepatic cells, our renal or kidney cells. All of these undergo mitosis. So if one of our somatic cells divides, it's going through the process of mitosis and produces two diploid daughter cells, which are abbreviated 2N. And both of these two daughter cells will have the identical chromosomes in both the identical numbers and the identical numbers of pairs of chromosomes as well. And so the members of each pair are called homologous chromosomes. Meiosis is a very special form of cell division, and it only occurs when we are producing gametes. So meiosis does not occur with our skin cells or our muscle cells or anything else throughout our body. It is only going to occur when we are producing sperm in males or oocytes in females. So the outcome of meiosis is that there will be 4 haploid gametes, which are abbreviated lowercase n, and each of these will have 23 individual chromosomes, not 23 pairs of chromosomes, which makes sense because our cells throughout our body, our somatic cells, will be diploid, meaning that they have the full complement of genetic material, 50% from our father and 50% from our mother biologically. Now this will produce 4 haploid gametes, right? So each of these has one individual set of chromosomes. So when the male gamete meets with a female gamete, it produces a zygote with 46 chromosomes or 23 pairs of chromosomes. Figure 28.8 shows you what happens during mitosis as compared to meiosis. So starting off at the beginning, we have a parent cell. So the parent cell here is a diploid cell, meaning that it has the full complement of genetic material. So it has one haploid set from the mother and one haploid set from the father. And so on mitosis, this is all of our somatic cells again. What happens is we'll have chromosome duplication. So we'll have 2 sister chromatids, which will then be able to separate out. So during the phase of anaphase and telophase, you can see that we end up with 2 distinct daughter cells. And so those daughter cells each are 2N, meaning they both have the full complement of genetic material from their maternal chromosomes and their paternal chromosomes. So that's why if you were to do DNA testing on skin cells, for example, you would be able to identify both maternal identity and paternal identity. Now, our meiosis is a totally different process. So what we have here is we'll have a breakdown that results in daughter cells down at the very bottom that are haploid. So that means that each individual daughter cell has 50% of the genetic information that you would expect of a somatic cell, which again is important because the gametes, again oocytes and sperm, each only have 50% of the genetic material, so that way they can come together to create a complete complement. In the process of meiosis, the chromosomes are duplicated and then condensed so that each chromosome has two chromatids, which are arms of the chromosome. And so through a process called synapsis, the maternal and paternal chromosomes will come together, and these four matched chromatids form a tetrad, which is meaning four. And crossing over is the process by which genetic material is exchanged. And it's an incredibly important process because what it does effectively is to increase genetic variation among offspring. And this is how it is that two parents can have multiple children, but through the process of crossing over, they are not all the same child genetically. They will all be completely different because of the crossing over of these specific genes. The process of meiosis includes two cycles of division. The first is called meiosis 1, and the second one, not so cleverly, is called meiosis 2. So during meiosis 1, we have prophase 1 in which the nuclear envelope disappears. And in metaphase 1, the tetrads are going to line up along the metaphase plate. And remember what's pulling them along that metaphase plate? Centrioles. And in anaphase 1, those tetrads will break up, with each daughter cell then getting two copies of either the maternal or the paternal chromosome. So then the maternal and paternal components will then be randomly assorted and independently distributed. This results in telophase 1, which ends when two daughter cells are formed, and each of these daughter cells has a unique combination of chromosomes. The first meiotic division is a reductional division because what it's really doing is reducing the number of chromosomes from diploid to haploid. Diploid being the 2N, which is 46 chromosomes or 23 pairs of chromosomes, to haploid, which is the lowercase n and just 23 individual chromosomes. So both cells are going to have 23 chromosomes with two chromatids each. Interphase is the very brief period in between meiosis 1 and meiosis 2. And during this time, not a terrible amount is occurring, and DNA is not being replicated at all. In meiosis 2, a cell will go through the process of prophase 2, metaphase 2, anaphase 2, and telophase 2, just as in meiosis 1, with a little bit of difference. So in anaphase 2, this is when the chromatids will separate, and in telophase 2, we'll have four individual haploid cells, each having 23 chromosomes. So meiosis 2 is an equational division, whereas meiosis 1 is a reductional division. So in meiosis 1, we're reducing the number of chromosomes from diploid to haploid, or 2N to N, or 46 to 23. And in meiosis 2, this is an equational division because we have our chromosome number completely unchanged. Spermatogenesis is the process by which sperm are produced. It starts around puberty and then continues past age 70. So interestingly, what this means is that men have the opportunity to reproduce well into their 70s, whereas women typically will lose their reproductive potential at menopause, which occurs sometime around the 40s or 50s and sometimes even the 60s. This complete process of spermatogenesis will take about 64 days and includes three important steps. The first is mitosis, the second is meiosis, and this finally then will result in spermiogenesis, which is the production of sperm. The first part of spermatogenesis are the cells of spermatogenesis, and these are known as spermatogonia, which are the stem cells of spermatogenesis. So these will divide through a process of mitosis, yielding two daughter cells. So one of these daughter cells will remain as a spermatogonium, and the second one is going to be the one that goes on to become a sperm. So it will differentiate into a primary spermatocyte. Primary spermatocytes then will undergo the process of meiosis, which is when they have that reductional division and then the equational division, and then form secondary spermatocytes. Next, the secondary spermatocytes will differentiate in spermatids. And so spermatids are the immature gametes that will eventually differentiate into sperm. Sperm will then finally lose contact with the wall of the seminiferous tubule and enter the fluid in the lumen. Figure 28.9 reviews for you the process of spermatogenesis. First, it starts with mitosis, in which case each division of a diploid spermatogonium will produce two daughter cells. One becomes a spermatogonium that stays in contact with the wall of the seminiferous tubule, and the second one is a primary spermatocyte, and it gets displaced and pushed towards the lumen. So this takes about 16 days or so to occur. And then meiosis 1 occurs. And meiosis 1, remember, is our reductional division. So during this point, we're taking these 46 individual chromosomes and then breaking them down into individual secondary spermatocytes that have 23 chromosomes each. So they each have a pair of duplicate chromatids. And that takes about 24 days or so. Then next, we have meiosis 2, in which case the secondary spermatocytes will go into this process, and it results in 4 haploid spermatids. So because we had 2 diploid cells at the end of meiosis 1, they get divided into the 4 individual haploids. And then they are able to move forward into the process of spermiogenesis after only a few hours of being in meiosis 2. So spermiogenesis is really just the process of maturation of sperm. And so each spermatid will develop into a single sperm. And this takes about 24 days. In the seminiferous tubules, we have multiple different things going on. We have the spermatogonia. We have spermatocytes, and they're all at different stages of meiosis. There are spermatids, and then finally we have sperm. But also we have these large nurse cells, which are known as Sertoli cells, that are there to help support the process of spermatogenesis. These Sertoli cells, or nurse cells, are there to play a very important role in spermatogenesis because they help to support the whole entire process. They do this by maintaining a blood-testis barrier to prevent toxins and other substances from affecting the development of sperm cells. They also support mitosis and meiosis. They support the whole process of spermiogenesis, which is the maturation of sperm cells. They secrete inhibin, and they also secrete an androgen-binding protein. Spermiogenesis is the very last step of spermatogenesis. So remember, it starts with mitosis, then there is meiosis 1, meiosis 2, and then finally we have spermiogenesis, at which point each of these spermatids will mature into a sperm. And so this involves major structural changes, as you can see in this image to the right, which is figure 28.11 in your text. At spermiation, a sperm will do two things. It loses its attachment to a nurse cell, which has been supporting it all along, and it goes off on its own, entering the lumen of the seminiferous tubule. The blood-testis barrier is an incredibly important function because what it does is it will isolate the seminiferous tubules from the general circulation. The nurse cells, or the Sertoli cells, are those that maintain this barrier, and they're joined by tight junctions, dividing seminiferous tubules into their constituent compartments. The outer compartment, that is basal, will contain the spermatogonia. However, the inner component, which is the luminal component towards the lumen, that's where meiosis and spermiogenesis will occur. Nurse cells are stimulated by two different hormones of the male reproductive system. The first is FSH, or follicle stimulating hormone, and the second is testosterone. So when nurse cells, or Sertoli cells, are stimulated, they will be able to promote the division of spermatogonia and also the meiotic divisions of spermatocytes. Nurse cells support spermiogenesis by surrounding and completely enclosing the spermatids, so that allows them to provide nutrients and chemical stimuli for development. So essentially they're feeding the spermatids as they develop. They also phagocytize cytoplasm that's shed by the developing spermatids. The Sertoli cells, or nurse cells, also secrete inhibin. Inhibin is a peptide hormone that responds to factors that are released by sperm, so inhibin will depress both pituitary function of follicle-stimulating hormone, or FSH, and also hypothalamic secretion of gonadotropin-releasing hormone, or GnRH. The regulation of follicle-stimulating hormone and gonadotropin-releasing hormone by inhibin is important because it allows for a negative feedback cycle to control the process of spermatogenesis. So therefore, what this means is that as the sperm production cranks up, the secretion of inhibin also goes up as well. Nerve cells also secrete an androgen-binding protein, abbreviated ABP, and it binds androgens. Clever name. Primarily testosterone. And this occurs in the luminal fluid within the seminiferous tubules. This is important for two things. It elevates the concentration of androgens, and it also stimulates the process of spermiogenesis. Also, a side note, the production of androgen-binding protein is stimulated by follicle-stimulating hormone. So once the sperm is developed, it will leave the epididymis and it's technically mature, but it's still immobile, meaning that it can't move. So in order to become motile, or able to actively swim, and fully functional, the sperm has to go a process called capacitation. And there are two steps in capacitation, which makes it capable. So the first is the sperm becomes motile when it gets mixed with secretions from the seminal glands. The second part of capacitation is when the sperm becomes capable of actually fertilizing an egg, and that is once the sperm becomes exposed to components of the female reproductive tract. The anatomy of a sperm is pretty straightforward. At one end, we have the head, and at the opposing end, we have the tail. In between, there are two specific segments. There is the neck, which is right next to the head, and then in between the tail and the neck is a portion in the middle, literally called the middle piece. Let's look a little more closely at each of the four constituent components of a sperm. The first is the head, and this is a flattened ellipse that has the nucleus and the chromosomes in it, which makes sense when you think about what happens at fertilization. The head of the sperm is what gets in first, and it has the most important part, the nucleus with it. The acrosome is a membranous component at the tip of the head, and in the acrosome are enzymes, and these are essential to fertilization because they help to break through that egg. Also, this is made of fused saccules of the spermatid's Golgi apparatus. The middle piece, which is in between the neck and the tail, is where all the mitochondria are. And so the mitochondria here spiral around the microtubules, and this is, of course, the source of ATP generation for the sperm to allow the tail to flagellate. And then lastly, at the distal end of the sperm is the tail. And the tail here is the only flagellum, or flagella plural, in the human body. So the flagellum is a whip-like organelle that allows the cell to be able to move from one place to another, and it has a complex corkscrew motion as opposed to a dog wagging its tail from left to right. This also makes sense if you think about when you're in the water swimming and you try to stay upright. You may have done something called the egg beater, in which your legs go in these circles opposing one another, and that helps to keep you upright. And so it's the same sort of idea here, that the corkscrew motion is what allows for the sperm to be able to go in a specific direction. Mature sperm have to be all business. So because they've got a job to do, we're going to cut out anything that isn't incredibly important to maintenance of the sperm and for reproductive purposes. So mature sperm will lack an endoplasmic reticulum, the Golgi apparatus, lysosomes, peroxisomes, and also cellular inclusions. So the loss of all these organelles reduces the size and mass of the sperm, which is effectively the equivalent of a professional swimmer shaving all of their body hair to reduce their friction in the water so that it can be as quick and slick as possible. So losing all these organelles reduces the size of the sperm and also their mass. So that makes it easier for them to get around. The sperm also have to be able to absorb nutrients on their own from the surrounding fluid. So fructose is going to be the primary source of nutrients for sperm. Next, let's look at the hormones of the male reproductive system that help to regulate reproductive function. So first off, we have the anterior lobe of our pituitary gland, or our adenohypophysis, and it releases follicle-stimulating hormone and luteinizing hormone. These are both released in response to gonadotropin releasing hormone. Gonadotropin-releasing hormone is synthesized and released by the hypothalamus and is carried to the pituitary gland by that hypophyseal portal system, which we just covered in the last chapter. And so the gonadotropin-releasing hormone then can be secreted in pulses in between 60 and 90 minutes, give or take, and these pulses will allow for the rates of secretion of FSH and LH to be controlled. And it also controls the rate of secretion of testosterone, which is also released in response to luteinizing hormone. Together, follicle-stimulating hormone and testosterone target the nerve cells within the seminiferous tubules. And those nerve cells, remember, are there to support sperm as they develop. So those nerve cells secrete inhibin, they secrete androgen-binding protein, and they also promote spermatogenesis as well as spermiogenesis. So again, inhibin is important because it inhibits FSH production in the pituitary gland, and this can then in turn suppress the secretion of gonadotropin-releasing hormone at the hypothalamus, which then will allow for a faster rate of sperm production, causing more inhibin to be secreted again. Luteinizing hormone is going to target the interstitial endocrine cells in the testes, and luteinizing hormone there will induce the secretion of two things, testosterone primarily, and other androgens. Testosterone is the primary androgen that's responsible for maintaining male sexual function. It essentially functions just like all other steroid hormones in that it circulates through the bloodstream, is bound to one of the two different types of transport proteins, and will diffuse across the target cell membrane, binding to a specific intracellular receptor. So the hormone receptor complex then binds to the DNA in the nucleus of the cell. The effects of testosterone are widespread throughout the body. First off, it stimulates spermatogenesis. It also maintains libido or sexual drive and related behaviors, which is one of the reasons why you see on television frequently now lots of advertisements for various testosterone supplements. There also has the effect of stimulating bone growth and muscle growth, which is one of the reasons why at puberty there is a big rush of growth for young men. Also, it establishes and maintains male secondary sex characteristics, which is something like, for example, a beard or facial hair or body hair. Also, it maintains accessory glands and the organs of the male reproductive system. In some specific tissues, testosterone is going to be converted into dihydrotestosterone, which is DHT, in these tissues. So some of this dihydrotestosterone will diffuse into the bloodstream and make up to about 10% of our circulating testosterone levels. Some of this will bind to the same receptors as testosterone, and some tissues will respond specifically to dihydrotestosterone instead of testosterone. Estradiol, while an important component of the female reproductive system, is also present in the male reproductive system as well. So it's found in very small amounts, which is about 2 nanograms per deciliter, in the plasma of adult men. 70% of the estradiol is formed by circulating testosterone, and it's done so by aromatase, which interestingly, by the way, is affected by chemotherapy because some of the drugs that people take for cancer treatment are aromatase inhibitors. There are also unknown reasons why the production of estradiol goes up in older men. One idea may be that there is a potential relationship between the production of testosterone and estradiol and their competing receptors. Before we get into the female structures of the female reproductive system, I just want to tell you a quick fun story about when I was working as a PA at Medical College of Wisconsin. I was doing a frozen section, which is essentially looking at tissue microscopically while the patient is still on the table, so that way the surgeon can determine the course of surgery, whether they need to resect more, for example, or not. So I received the specimen and I cut into it, and typically the specimens I would receive would be in formalin, but these specimens aren't, so they're a little bit more slippery. Anyway, I cut myself is the long story short. So because the specimen was a breast tissue specimen from a considerably older lady, I figured, well, it's probably not a big risk for anything in particular. So I did notify occupational health and went home. And I got a call Saturday morning at 6 a.m. from the head of occupational health telling me that the patient tested positive for HIV and now I needed to take HIV prophylaxis. Anyway, long story short, the first test that was done showed that the patient was positive for HIV, and then the follow-up test, which was the Western blot, indicated that the ELISA was actually incorrect because the patient was undergoing chemotherapy of aromatase inhibitors, which could indicate a false positive on an HIV test. Anyhow, so fortunately I was able to stop my prophylaxis and I was not exposed, but nonetheless, when you are working in healthcare, please be ever so careful. Next, let's look at the female structures of the female reproductive system. The female reproductive system is responsible for producing sex hormones as well as gametes. And so the female reproductive system will function to protect and support the developing embryo while the embryo is in utero. It nourishes the newborn infant via lactation. And the main organs of the female reproductive system are the ovaries, where ova are produced, the uterine tubes, which is the passageway by which they're carried to the uterus, which is the primary housing for a developing embryo and fetus, the vagina, which is the space by which the embryo or fetus would pass at delivery, and the external genitalia. Figure 2813 shows you the entire female reproductive tract in cross-section. So what we're looking here is a cross-section through the pelvis, so you can see the right half here specifically of the female reproductive system. So up at the top, what you see is there is an ovary with the fallopian or uterine tube wrapped around it. You see there's almost like a hand that reaches around that, and those are the fimbria of the uterine tube. So the uterine tube is in direct connection with one side of the uterus, which you can't see here because this is right through midline. So that connects with the uterus, which is that large C-shaped structure that you see in the center of the image. So you can see this uterus there sort of dips down into the very top of the vagina, and there are these little crevices up towards the side on the left and the right, or I guess here in this image is really the anterior and the posterior aspect of the uterus, and that's called the vaginal fornix. So then there's the cervix, which is the most bottom component of the uterus, and then just inferior to the cervix we have the vagina, and then anterior to that you can see there's the bladder as a large resting space, and then the urethra that goes down. So in the female reproductive system, the bladder and the urethra are not involved. However, the urethra is involved in the male reproductive system because it is a conduit for both semen and urine. But in the female system, the urethra is only a conduit of urine. And then posterior to the vagina, you'll see then there's the anus, the rectum, and the rest of the GI tract, which is unrelated, but I just wanted you to be able to identify what you're looking at here. There's a broad sheet of material that encloses the ovaries, the uterine tubes, and the uterus, and this holds them all in place so they're not just wobbling around all over the place. And this is called the broad ligament because it is very wide and very broad. So you'll notice here that if you look closely, the uterine tubes up at the top on both the right and the left run alongside the broad ligament, and then those uterine tubes will open up into the pelvic cavity just lateral to the ovaries. So what happens is they open up as if you were to imagine in person, I would stand up in front of you and demonstrate this with my arms. So I would make my body into the uterus, and my arms are going to be the uterine tubes. So I'm stretching my arms out laterally to either side, and I stretch my fingers out because my hands are the fimbria. So if you can imagine that the arms are hollow, and that's where the lumen is by which the egg would travel. At the hand, at the palm, there's the equivalent of an opening. And so those fimbria or fingers of the fallopian tube literally will grab onto the ovary and absorb the egg as it's being expelled from the ovary. The female reproductive system has two little spaces called pouches that are just little bits of space, really. But these little bits of space are helpful because if you're passing an embryo or a fetus through the birth canal, every little bit of space that can be compressed and stretched helps. So there is one that is anterior and one that is posterior. They are basically in relationship to the uterus. So the one that is posterior is the recto-uterine pouch, so it's in between the uterus and the rectum. And then the one that's anterior is the vesico-uterine pouch, which is in between the bladder and the uterus. So the image that I'm showing you here on the right is an image that a graduate student of mine had taken back when I was working at Rosalind Franklin University as a professor there. And what this is showing is a case of extremely advanced cervical cancer. So let's orient ourselves just for a moment. So this is the image of a woman standing as if she is facing towards the right side of the screen. And so she's facing that way. So as you can see on the left side of the screen, we have the large structure being the rectum and anus. And then on the anterior aspect, what's cut off is a portion of the bladder. What's dead smack in the center of the image is this area of white discoloration, and that is actually cervical cancer that has managed to invade a number of structures. So at the very top in the center of the image is the uterus, and so this would actually be the left half of the uterus. And what we're looking at is an otherwise normal uterus, but, however, at the cervix, all of that white, that is all-invasive carcinoma. And so everywhere that you see white, you can see it coming down the posterior wall of the vagina, the anterior wall of the vagina. That cervical cancer is invading both anteriorly to the bladder wall by the vesicouterine pouch, and it's also invading posteriorly by the rectouterine pouch and invading the wall of the rectum as well. The ovaries are the small organs that are almost almond-shaped, and they're on the lateral walls of the pelvic cavity, right next to the fimbria of the uterine tube. The ovaries have three main functions. The first is that they produce immature female gametes, which are oocytes. They also secrete female sex hormones, or these are estrogens and progesterone. And they also secrete inhibin, which is involved primarily in the feedback control of pituitary follicular stimulating hormone. So the image on the right is a gross image of a absolutely normal looking ovary. So what you see at the top, that big yellowish sac, that is a corpus luteum. So that yellow area is all full of those steroid sex hormones because it's full of lipids. And that is there to help develop the egg as it develops. You'll also notice in this image that there are lots of little white spots, and those white areas are not malignancy, they are totally normal structures. And what they indicate is these are just corpora albicantia, which are the bodies of those corpora lutea as they've basically devolved and are no longer needed. So after they've served their purpose, they will basically devolve into these little fibrotic, tiny little areas that will eventually become absorbed back into the stroma or the tissue of the ovary. The ovaries are supported by the mesovarium, the ovarian ligament, and the suspensory ligament. So all of these together keep the ovary in place so it doesn't twist or flop or bend upon its blood supply. So the mesovarium is a thickened fold of mesentery that you can see in this image just in between the uterine tube and the ovary on the superior aspect of the ovary. And it supports and stabilizes the position of each ovary. There's also the ovarian ligament, and you can see in the image here that the ovarian ligament stretches from the ovary itself to the body of the uterus. And then there's the suspensory ligament, which sort of dangles from the ovary to the pelvic wall. And this contains the ovarian artery and the ovarian vein. So as you can imagine, the suspensory ligament needs to stay in place. If it were to become cramped or bent, it could cause a lack of blood supply to the ovary. So these vessels are all going to connect to the ovary at what is called the ovarian hilum. And the hilum is the point where all the blood vessels typically enter and exit a major organ. And so the ovarian hilum is where the ovary attaches to the mesovarium at the most superior aspect of the ovary. The ovary is covered by a visceral peritoneum called the germinal epithelium because this is the site at which we're going to have eggs develop. So this covers the surface and overlies the tunica albuginea, which is a whitish covering that's consisting of columnar epithelial cells. The stroma is going to be the background tissue that is interior to the ovary. And within the stroma, there are two components, the cortex and the medulla, which is also just like the adrenal gland or just like the kidney or a number of other organs. So the cortex is the outer aspect and the medulla is more towards the center. So the gametes are going to be produced in the cortex around the periphery or most superficial aspect of the ovary. The uterine tubes are also called either fallopian tubes or oviducts, and these are just hollow muscular tubes that are about 13 centimeters long, and they will transport the oocyte to the uterus. So there are three segments of the uterine tube, the infundibulum, the ampulla, and the isthmus. So the infundibulum is the expanded component that's a funnel near the ovary. So it's opened up because this is where the hands are to be able to grab on to the ovary itself. So the fimbria are the finger-like projections that extend into the pelvic cavity or onto the ovary. And the inner surfaces of the infundibulum is lined with cilia. And the cilia here is very important because what do cilia do? They wave. So they are waving the ovum along its way throughout the uterine tube. The ampulla is the middle segment of the uterine tube, and its thickness is going to increase as we get closer to the uterus. And then the isthmus is going to be that little short segment that's right in between the ampulla and the uterine wall where the two meet. As we've discussed in so many other lectures, our structure determines function. So if we have the function of the fallopian or uterine tube being to serve as a conduit for an oocyte to travel from the ovary to the uterus, how would we make that happen if we were designing the structure? Well, here's how we look at it. There are basically three ways that we could move something along in a tube. One would be peristaltic contractions, such as the way food is propelled through our gastrointestinal system. Another would be having a flagellum or flagella plural, such as the case of sperm. And the third would be ciliary movement, which can help to wave an object along. So within the uterine tube, we do not have flagella, of course, but we do have the two other mechanisms of moving something along. There is ciliary movement in the inner uterine tube wall that helps propel the oocyte along. There are also peristaltic contractions that can occur as well. So the anatomy of this is such that if we're going to have cilia, where are they on? Well, we have ciliated columnar epithelial cells that line the stellate or star-shaped lumen, which is just the opening of the tube, basically. And that lines the entire mucosa. So the mucosa is surrounded then by concentric layers of smooth muscle, which are also going to be involuntary and can also be stimulated by prostaglandins. So if we look at the image on the right, you're going to see at the bottom a light microscopy image of a cross-section of a uterine tube showing you the stellate or star-shaped lumen with those ciliated columnar cells and occasional mucin-secreting cells. The image above that, I think, is fascinating because it's a scanning electron micrograph image at 4,000 power looking at a colored view of the epithelial surface of the fallopian tube. And so you can see all those cilia sticking up along, and they will wave that oocyte along. As children, I think we all probably learned that when an egg meets a sperm, they implant in the uterus, and then it grows for the next 9 months. However, it's a little bit more complicated than that, and it's actually very timing specific. So an oocyte, specifically a secondary oocyte, has to meet a sperm within the first 12 to 24 hours of its being released from the ovary. And so when it meets, it's actually going to meet in the uterine tube and then travel along and finally implant in the endometrium of the uterus. So technically, this means that fertilization occurs in the uterine tube near the boundary between the ampulla and the isthmus. So within the epithelium of the uterine tube, we also have something called PEG cells. And PEG cells are there to secrete fluid that does two different things. It supplies nutrients to the sperm and the pre-embryo, and it also helps to complete capacitation of sperm. The uterus provides the housing for an embryo that's developing, and the embryo is defined as being in weeks 1 through 8, and a fetus, which is week 9 through whatever point delivery arises. So the uterus is going to do a few things. It's going to provide a mechanical protection for a developing embryo and fetus because it is a thick muscular wall. It also provides nutritional support via the placenta, and it also, via the placenta, provides for waste removal for a developing embryo or fetus. So as you can see from figure 2815, the overall general shape of a uterus is an upside down pear, basically. So it's about 7.5 centimeters long, 5 centimeters in diameter, and weighs about 50 to 100 grams. What this really means functionally is if you're a lady with small hands and you pull your hand together into a tight fist, it's about that size. In fact, it's probably even smaller than that. So it's kind of fascinating that this is the organ that can blow up to hold up to a 10-pound infant. Now, it normally bends slightly anteriorly near the base, and that's called anteflexion, although there's a number of different times where women will have retroverted or retroflexed uteri that bend slightly backwards. An interesting clinical correlation, the degree of flexion, for the most part, provided it's not severe, has no real factor in anything, whether it's in sexual intercourse or in labor and delivery. But the one thing it does affect is how you experience cramps. So if you have a more anteflexed uterus, it's possible to have more abdominal cramps, whereas if you have a more retroflexed uterus, when you have cramping, it's likely to be more in your lower back. So by the way, the image on the right, this is figure 2815. I have to tell you, you guys are all probably far too young for this, but when I was younger, I look at this picture and I think of a show called The Wonder Years, in which they learned in sex ed that this looked like a cow with earrings, which I think I'm going to always keep in my mind forever every time I see this image. The uterus is held in place by a number of different ligaments that support it so it doesn't just twist upon itself, flip over, or do any weird acrobatics. So the first is the uterosacral ligaments, and these are there to prevent inferior/anterior movement. The round ligaments are also present, and they're present laterally when what they do is they're going to restrict posterior movement. So they extend through a very small inguinal canal right along with the ilioinguinal nerves. And then lastly, we also have the cardinal ligaments, which are kind of difficult to see here, but if you look at the image on the right, the bottom of the image is the anterior, the top of the image is the posterior. So we're looking down into the pelvic cavity from above, as if the person is facing us. And so what you'll see is posteriorly, you can see the uterosacral ligament, and then also you can see that the cardinal ligaments are actually underneath the broad ligament, and they're kind of difficult to see, but they're those thinner looking, more broad ligaments present. Let's look in a little more detail at the uterine body. So the uterine body is the majority of the uterus, but the part of it that sort of sticks up as a rounded top part, if you look at the very center top of the image on the right, is the fundus of the uterus. And so it is superior to where the uterine tubes approach. Now, the uterine body comprises the majority of the uterus, so it extends from the fundus, which is where the uterine tubes come in, all the way down to the cervix. And so it's going to stop basically right at the isthmus of the uterus, which you can see is labeled on the left here, and it is just at the level of where the cervix begins. The cervix is the most inferior component of the uterus, and it extends from the narrowed part, the isthmus of the uterus, into the vagina. So the distal end of it is actually extending about 1.5 centimeters into the vagina, and you can see at the most inferior aspect there's something called the external os. And so the os literally just means mouth. So for example, if you're ever told before surgery your NPO, that's nil par os, meaning nothing by mouth beforehand. So in the same case, this is the os, or the mouth of the uterus, and is technically the external os of the cervix. So this is the opening, and it leads up into the cervical canal. So the cervical canal is going to be a passageway that's quite rigid and rubbery, because it's got a lot of connective tissue and a lot of collagen fibers, and this is a constricted passageway that allows opening to the uterine cavity via the internal os. So if you look at this image here, at the very beginning of the cervix, right down at the most inferior aspect, that is the external os, and then there's a slight dilatation, and then you see that it constricts just a tiny little bit again, and that second constriction is the internal os. The uterus is supplied with a lot of blood supply via the uterine arteries, which you can see here coming in on the left side of the image, and there's also accompanying uterine veins. So these arise off of branches of the internal iliac arteries. And the ovarian arteries, interestingly, come off of the abdominal aorta. Now there are all sorts of other veins, lymphatic vessels around. As you can see, there's multiple sources of vascularity. And here's another clinical correlation for you. So previously, when women have been diagnosed with fibroids or leiomyoma, which are just smooth muscle tumors that are benign, that occur very commonly in the uterus, we would usually treat that if it was symptomatic by either trying to resect a specific leiomyoma, which can be tricky because they're typically intramural or within the wall of the uterus, or we would treat it with a hysterectomy. But now there's a procedure that's abbreviated as a UFE, or UFE, which is a uterine fibroid embolization, in which case the blood supply to the specific tumor can be reduced and cut off entirely, so that way it involutes and eventually gets reabsorbed by the body without requiring surgery. The uterus is innervated by autonomic fibers that arise from the hypogastric plexus, which is sympathetic, and also from sacral segments S3 and S4, which provide parasympathetic innervation. Segmental blocks are something that is typically done during labor and delivery, which is an anesthetic procedure, and so they will try to target spinal nerves T10 through L1 at this point. The uterine wall is comprised of three main layers. There's the outer aspect, which is the perimetrium, the middle layer, which is the myometrium, and then the inner layer, which is the endometrium. So the perimetrium is an incomplete serous membrane that's continuous with the peritoneal lining. And it's going to cover the fundus and the posterior surface of the uterine body, as well as the isthmus, and honestly, a component of the anterior uterus as well. The myometrium is the muscular layer, so myo meaning muscle, so it is the thickest component of the uterine wall. And then the endometrium is probably the most relevant in our lives because endometrium is both where a fertilized egg would implant, as well as the lining which sheds monthly as part of a monthly cycle. So the inner endometrium is very thin and it's a glandular layer. The myometrium is the thickest portion of the uterine wall, and it comprises the vast majority of it. In fact, almost 90% of the entire mass of the uterus. The muscular layers of the myometrium are arranged in three different orientations, longitudinally, in a circular fashion, as well as obliquely. And this is specifically designed to be able to provide force so that way it can push the fetus down and out of the uterus into the vagina during labor and delivery. The image that we're looking at on the right is a bivalved uterus, or a uterus cut into halves, usually front and back, anterior and posterior. And what we're looking at here are multiple leiomyomata, or fibroids, which are benign tumors, and these all look totally normal to me grossly. But as you can see, there's lots of them. They're everywhere. These are very common in women, and furthermore, they get in the way with two different times. One is during labor and delivery, of course, and the second would be as part of a monthly menstrual cycle. Cramping would cause considerably more pain and pressure because of these nodules or tumors in the way. The endometrium is the inner lining of the uterus, and it contributes only about 10% of the mass of the uterus. So between the myometrium and the endometrium, that means the perimetrium is really just the outer lining and really doesn't do a whole lot. So the endometrium is the glandular and vascular tissue that supports the demands of the growing fetus physiologically. There are many, many uterine glands present here, and they all open up into the endometrial surface, which is in the endometrial cavity. And these are going to extend deep into the lamina propria. Estrogens here are going to cause the uterine glands, the blood vessels, and the epithelium to change monthly as part of a cycle. The image that you see on the right is a cross-section of uterus. And what I'd like to draw your attention to is the pink part in the center over to the right. That's not any lesion of any sort. All that is because it hasn't been fixed fully in formalin, which is formaldehyde essentially, yet. So because it's not fully fixed, we have this discoloration in the center, which is really just what live tissue looks like. Everything that's been fixed in formalin starts to take on this tan gray looking appearance. What I'd like to draw your eye to is the textural difference between the top and the bottom of the myometrium over at the far right of the image. What you'll see is at the top, there's that tan white homogeneous thickened area. That is endometrial carcinoma. And you can see this kind of tricky to say where it starts and stops along that pinkish myometrium, which is totally normal. Those are normal blood vessels and glands that are present within the myometrium itself. So we actually estimate the invasion of endometrial carcinoma based on whether or not it is 50% or greater of the depth of the myometrium. And that delimiting factor of whether it's reached the threshold of 50% of the total thickness of that wall will determine whether chemotherapy or radiation is required following surgical resection. The endometrium has two distinct layers, the basal layer and the functional layer. So the basal layer, like its name, is at the base of the endometrium, attaching the endometrium to the myometrium below it. And so the basal layer will have terminal branches of tubular uterine glands, which we'll look at in just a moment. Now the functional layer is the more superficial of the two layers of the endometrium. And the functional layer is the one in which most of the uterine glands are present. So this contributes to the majority of the thickness of the endometrium, which ranges from just a couple of millimeters all the way up to maybe 5 or 6 millimeters when it's at full thickness. And this undergoes dramatic change in thickness and structure during the uterine cycle as the functional layer builds up and then sloughs off in a menstrual period. The functional layer of the endometrium is the one that undergoes the cyclical changes, producing the main characteristic of our uterine cycle. So the functional layer responds to varying levels of sex hormone levels. The basal layer, however, does not really undergo such considerable change. So if we look at the image on the bottom, you'll see at the right, labeled B, a light microscopy image of the uterine wall. So at the leftmost aspect of this image, you're going to see the uterine cavity. So that's the superficial endometrium. And then as you proceed over towards the right, you'll see what's labeled at the right is the myometrium, which is the muscular wall of the uterus, which comprises the vast majority of the bulk of the uterus. The perimetrium isn't even seen here in this image. So you'll notice that on the left, you'll see that in the functional layer, we have many different uterine glands. But as we move deep into the basilar layer, you're going to see that there's very few glands. And in the myometrium, of course, there aren't any glands really that are notable. What we have are the 3 different layers of smooth muscle tissue. The endometrium is supplied by several different arteries. The arcuate arteries are those that encircle the endometrium, and the radial arteries will supply both the straight arteries, which support the basal layer, and then they also supply the spiral arteries, which supply the functional layer. So it's the spiral arteries which have the greatest effect on those specific functional layer monthly, as the functional layer is the one that builds up and sloughs off. The vagina is an elastic muscular tube which extends between the cervix and the vestibule, which is the opening at the labia. So commonly people refer sometimes to female external genitalia as the vagina. So please make a note of that. So when you're doing histories and physicals, if somebody complains about pain in their vagina, follow up to be sure that they are specifically referring to the vagina as opposed to the potential labia. So the vagina extends between the cervix and the vestibule. It's about 7 to 9 centimeters long and is highly distensible, meaning it can stretch out considerably in all 3 different dimensions. It has 3 functions. The first is for passageway of menstrual fluids as part of the monthly cycle. There's also the purpose of having sperm to be able to pass through here, through the cervix during sexual intercourse. And then the third is it actually forms the most inferior portion of the birth canal. So as a fetus would be delivered through the uterus, through the cervix, it would also pass through the vaginal canal before entering the external world. The cervix actually projects inferiorly into the vaginal canal, creating the vaginal fornix, which is that shallow recess that's surrounding circumferentially the cervical protrusion. So this is where the cervix actually goes up around the lip, basically, of the cervix. So this lies parallel to the rectum posteriorly and anteriorly to the urethra. On either side of the vaginal entrance, there are 2 bulbospongiosus muscles, and these contractions will constrict the vagina and cover the bulb of the vestibule. So this is effectively a mass of erectile tissue that's on each side, and it has the same embryonic origins as the corpus spongiosum of the penis. So basically what this means is the corpus spongiosum would not be able to develop in males if this specific tissue did not also exist in females, because they both arise from the same embryonic tissue. The hymen is an elastic fold of epithelial tissue that partially blocks the entrance to the vagina. It's usually torn by things like sexual intercourse, tampon use, or pelvic examination, but, and this is the most important part, it's also disrupted by physical activity. So historically, when women were evaluated for virginity by looking at their hymens to see if it was intact, we know now that that's not exactly a great measure, because just running around, playing soccer, being a normal little kid, all of these things will cause disruption to the hymen to some extent. The vagina is supplied with blood through the vaginal branches of the internal iliac artery, or the uterine arteries and veins. And the vagina is innervated by 3 different places: by the hypogastric plexus, the sacral nerves at levels S2 through S4, as well as branches of the pudendal nerve. Let's look at the histology of the vagina. The vaginal wall has a network of blood vessels and multiple layers of smooth muscle, and this is moistened by secretions from the cervical glands, as well as water movement across the permeable epithelium. So the image on the right shows you a light microscopy image of vaginal epithelium. So you can see in the far left, that would be the lumen of the vaginal canal, or the most superficial component of the vaginal canal. And then the dark purple part that we see is the epithelium. And you can see the darkest purple is that irregular line at the basement membrane of the epithelium as it meets the lamina propria. And then you can see there are multiple blood vessels within the smooth muscle just subjacent to the epithelium. The vaginal epithelium is a non-keratinized stratified squamous epithelium, and it forms folds which are called rugae. So you can see when we look at the image on the right, you can see the rugae are listed about in the middle of the image, showing you how there are multiple folds in the vaginal epithelium. The vaginal lamina propria is thick and elastic, and in the lamina propria, we have multiple small blood vessels, nerves, and lymph nodes. The vaginal mucosa is also surrounded by an elastic muscular layer, and this muscular layer is comprised of multiple layers of smooth muscle fibers arranged in both circular and longitudinal bundles. So this is continuous with the musculature of the uterine myometrium. Within the vagina, there are multiple different bacteria populations that live, and these are harmless residents of the vagina. So they cause no problems in and of themselves and actually are part of the normal flora of the vagina. So these are supported by nutrients in the cervical mucus and create an acidic environment, which is important for producing an environment in which not many pathogens can live. So interestingly, by the way, a new practice that's being done in labor and delivery is if a woman doesn't pass a baby through the vaginal canal at delivery, so for instance she has to have a cesarean section, one of the things that's being done is something called the gauze method, by which a piece of gauze is inserted into the vagina to collect a sample of the fluid that's present in the vagina, which houses this resident bacteria, and then that is actually smeared in the infant's nose and mouth to be able to give them the exposure to those same bacteria and microorganisms that they would have had exposure to should they have passed through the birth canal. This is supposed to have an impact on their future immune function, and there is new burgeoning research on this topic, which is definitely worth looking into if you're interested. Vaginitis is an inflammation of the vagina, and it's caused by a number of different microorganisms ranging from fungi to bacteria or even parasites. So what this means is a diagnosis of vaginitis is nonspecific. Also, there's something called a vaginal smear, in which you can take a sample of epithelial cells at the surface of the vagina, and this can be used to look at the stages of the ovarian cycle as well as the uterine cycle. Looking at the external genitalia, the largest structure you'll see is the vulva or pudendum, and this is the area that contains the female external genitalia. Then there is the vestibule, and the vestibule is that central location that is bound by small folds, which are called the labia minora, or small lips, as opposed to labia majora, which are big lips. And the labia minora and vestibule are covered with smooth, hairless skin. The urethra will open up into the vestibule, and this is just anterior to the vaginal entrance. So in the image to the right, it actually is superior because this is an image of a woman in stirrups as opposed to a woman standing in anatomical position. The urethral glands will discharge into the urethra near the external opening. The clitoris is a small projection in the vestibule, which you can see at the most superior portion of this image here. And it's derived from the same embryonic structure as the penis is, so it has erectile tissue in it. So the corpus cavernosum of the clitoris is what forms the body of this. There's a small erectile gland that sits right on top of the body, as well as extensions of labia minora which form the prepuce. Also present with the vestibule are the lesser vestibular glands and the greater vestibular glands. So the lesser vestibular glands will secrete their product onto an exposed surface of the vestibule, and the greater vestibular glands will secrete their product into the vestibule near the vaginal entrance during sexual arousal. The mons pubis and labia majora can be discussed together, and they basically form the outer aspect of the vulva. So the mons pubis and labia majora will protect and cover the inner structures. These contain adipose tissue, and they're also covered with coarse hair. There are also sebaceous glands present, as well as apocrine sweat glands, and these secrete onto the inner surface of the labia majora. Breasts are known as mammary glands, and these are specialized organs within the integumentary system, which is our skin. These mammary glands then will produce milk through a process called lactation to nourish an infant. Lactation is a process that is controlled by hormones, and overall the development of mammary glands is also controlled by hormones. These lie in the pectoral adipose tissue. The nipple is going to contain ducts of mammary glands, and the areola is that reddish-brown, slightly hyperpigmented skin that circumferentially surrounds each nipple. So if you look at the image on the right, what you'll notice when we look at the general anatomy of the breast is that the pectoralis major is the primary muscle which is posterior to the adipose tissue, which contains in it the lobules of the mammary gland, the ducts, and you can see that the ducts all run through the nipple and out. The mammary glands will consist of lobes, and each of these lobes will have several secretory lobules separated by dense connective tissue. The mammary gland ducts will then be able to leave the lobules, and they converge or come together to form a single lactiferous duct in each lobe. The lactiferous duct is going to form an expanded chamber, which is called a lactiferous sinus, and there's about 50 to 20 of these each that open into the nipple. Within the breast, the suspensory ligaments are bands of connective tissue that originate in the dermis of the overlying skin, and they extend down into the breast tissue. So the areolar tissue then will separate the mammary gland complex from the underlying pectoralis muscles, which we see here as posterior to the breast tissue itself. The blood supply of the mammary glands is supplied by branches of the internal thoracic artery. Mammary glands can be considered to be either active or inactive, and the active mammary glands will be composed of a tubuloalveolar gland that has multiple glandular tubes, all ending in secretory alveoli. Now, something important to note here is that the breast tissue here, specifically the secretory apparatus, doesn't complete its development unless a pregnancy occurs. So we have a permanent histologic change that occurs via pregnancy. What we're looking at here at the bottom is on the left, we have a light microscopy image of a resting mammary gland, and you can see how purple the image is. So we've got these secretory alveoli that are dense and ready to go. And on the right, you're going to see a very different looking histology. An active mammary gland here has all of these glands, the secretory alveoli, opening up with a lot of milk present being produced in these alveoli. So you can see all that pale pink homogeneous looking material. That is all milk that's been produced. Onwards, let's look at section 28.5, oogenesis, and the female hormones that control the female reproductive system. Oogenesis is the process of producing ova, or singular ovum. So oogenesis begins well before birth, and the process of this accelerates at puberty, but it has a full hard stop at menopause. So the fetal ovaries will contain numerous oogonia, which are the female reproductive stem cells, which will then undergo mitosis, producing diploid primary oocytes. So these fetal ovaries will have about 7 million primary oocytes after 5 months of development, and these are all of the oogonia that we will ever need. So at birth, the ovaries will contain about 2 million primary oocytes. Most of these will degenerate in a process that is called atresia. However, those that survive will remain in prophase 1 until puberty. Around puberty, though, we'll have about 400,000 primary oocytes remaining, which is still a tremendous number, and some of these will be stimulated to finish meiosis 1, which will then produce our haploid secondary oocyte. In oogenesis, the cytoplasm of the primary oocyte will divide unevenly. So it will produce one secondary oocyte that has most of the original cytoplasm, and it produces 2 or 3 polar bodies, which are just these little ancillary side guys that will disintegrate. So the ovary will then release the secondary oocyte, which is in a suspended stage of metaphase 2. And meiosis 2 is only completed upon fertilization, and at that point, then we have a mature ovum that is formed. The primary oocytes will remain in suspended development up until puberty. However, at puberty, hormones change, and therefore the rising follicle stimulating hormone will trigger the start of the ovarian cycle. And every subsequent month thereafter, up until menopause, some primary oocytes will be stimulated to develop further. Let's take a look at the process of oogenesis as a schematic. So starting off at the top, we start with mitosis of oogonium. So we have our oogonium, which is our stem cell, and it can either become an oogonium or it can become a primary oocyte, which is diploid, by completing the mitotic divisions before birth. And then, through the process of DNA replication, in between the 3rd and 7 months of fetal development, these daughter cells here, the primary oocytes, will get ready for meiosis. And so as they do that, they're preparing, and they get as far as prophase 1 of meiosis 1, so the very beginning, but then it comes to a stop. So at this point, the primary oocytes are just going to hang out in suspended development until puberty. At that point, the FSH rising levels will trigger the start of an ovarian cycle. Subsequently, each month that there's an ovarian cycle, some of those primary oocytes that we see here in this second level will be stimulated to go into further development and complete meiosis 1, which yields the first polar body and a secondary oocyte. So if we look over to the right here, you'll see the secondary oocyte is the large structure, and the first polar body is just that little side guy that has some junk genetic material for the most part. And then we have meiosis 2. So each month after this, once puberty occurs, the ovarian cycles will produce both a second polar body, and they'll also work towards producing a mature ovum, which isn't actually generated until the time of fertilization. Ovarian follicles are specialized structures that are in the cortex, or the outer aspect of the ovaries, and this is where oocytes grow and where meiosis 1 will occur. The primary oocytes will be located in the outer aspect of the ovarian cortex near the tunica albuginea, which is that whiter outer aspect, and they're in clusters called egg nests. We begin the process of oogenesis with primordial follicles that are in egg nests. So these are located in the outer aspect of the ovarian cortex near the tunica albuginea in those little egg nest clusters. There's a single layer of follicle cells that surrounds each of the primary oocytes within the egg nest. Between the 3rd and 7 months of fetal development, the formation of primary ovarian follicles begins. So this can almost take up to a year to complete on occasion. But this begins when the primordial ovarian follicles are activated and they become primary ovarian follicles. So you can see here now that we have microvilli from the surrounding follicular cells, and these microvilli are going to originate at the surface of the oocyte. So that area is called the zona pellucida, or the clear area. The secondary ovarian follicles arise from the primary ovarian follicles, but not at a 1-to-1 ratio. Only some of the primary ovarian follicles will become secondary ovarian follicles. Next, we have tertiary, or the 3rd ovarian follicles. And in the tertiary ovarian follicles, you can see that there's a lot of follicular fluid accumulating in between the inner and the outer cellular layers. And the tertiary ovarian follicle is actually double the size of the secondary ovarian follicle. Now that the tertiary follicles are ready to complete their maturation, we can begin the ovarian cycle. So the ovarian cycle is a 28-day cycle on average. It can be as few as 20 and as many as 45, but it's typically 28 days, which is why most oral contraceptive pill packages have 28 days. By the way, some of them only have 21, and that's because there are 21 active hormonal pills, and then they give you 7 placebo or sugar pills just to keep you in the habit of taking the pill every day through that last week. But this is the monthly process of maturation, ovulation, and then degeneration of the tertiary ovarian follicle. So we're going to break down the ovarian cycle into 2 phases, the follicular phase and the luteal phase. The follicular phase is also known as the pre-ovulatory phase, and during this phase, the tertiary ovarian follicle will create a bulge in the wall of the ovary at the cortex. At this point, the oocyte and the follicular cells around it are going to project into the antrum, which is the central chamber of the follicle. These are surrounded by granulosa cells, and there are cells in the adjacent ovarian stroma that are there to form a layer of thecal endocrine cells, and these thecal cells produce estrogens. The rising luteinizing hormone levels are going to prompt the completion of meiosis 1, and the corona radiata are granulosa cells that remain associated with a secondary oocyte. Ovulation occurs when a tertiary follicle releases a secondary oocyte into the pelvic cavity, and then we have the fimbria of the uterine tube which are able to grab that. So the oocyte moves into the uterine tube by contact with the fimbria or even fluid currents, and the ovarian follicles that started but didn't get all the way through the process will now degenerate in a process called atresia. The luteal stage, which is the post-ovulatory phase, is one in which the corpus luteum, which is the large yellow-orange structure that you see up top here, is forming from the remaining granulosa cells under stimulation from luteinizing hormone. So the reason that's so yellow is because that yellow is due to the presence of cholesterol, and cholesterol, as we know, can convert into a number of different sex hormones. Specifically, it gets converted into progesterone. And this is very important for preparing the uterus for pregnancy and maintaining a placenta throughout pregnancy. So the progesterone produced by the corpus luteum is going to secrete a moderate amount of estrogen as well, and it will start to degenerate about 12 days after ovulation if fertilization doesn't occur. Also in the luteal phase, we have the development of corpus albicans singular, or plural, corpora albicantia. And so corpora albicantia are produced by these non-functional corpora lutea, or individual corpus luteum, as fibroblasts invade and form scar tissue. So the development of a corpus albicans is what marks the end of the ovarian cycle. So as you can see in this image on the right, everything that you see here that's white is going to be a corpus albicans. So I think probably the very best example of this is if you look at the furthest most right component of the image, you'll see what looks like a really great convoluted looking corpus albicans. That's a perfect example of one. The uterine cycle is also known as the menstrual cycle, and this is a repeating series of changes that occur within the endometrium. Your textbook says that it lasts between 21 to 35 days, averaging 28 days. However, it can extend up to as long as even 45 days and be within normal limits. Menarche is the first uterine cycle that occurs during puberty, which typically occurs around age 11 or 12. However, it can occur earlier. Menopause is when these uterine cycles terminate, and this can be expected to occur somewhere between age 45 and 55. The uterine cycle is broken into three phases: the menstrual phase, the proliferative phase, and the secretory phase. So the menstrual and the proliferative phases will occur during the ovarian follicular phase, and the secretory phase coincides with the ovarian luteal phase. During the menstrual phase, the endometrial functional layer, not the basal layer, will degenerate. And so this occurs in a patchy format, which leads to menstruation, or menses. And this is caused by constriction of the spiral arteries that support the functional layer of the endometrium. So the spiral arteries are going to reduce blood flow and therefore oxygen and nutrient supply to the endometrial functional layer. And then the arterial walls rupture, and as that occurs, then that releases blood into the connective tissues of the functional layer. During menses, the entire functional layer throughout the endometrium is lost, and this is shed through the external os of the cervix and the vagina. So only the functional layer is affected here by menses. The basal layer, which is deep to the functional layer, is supplied by the straight arteries and is therefore unaffected by this process. Menses can last between 1 to 7 days, and during this time, a range of between 35 to 50 milliliters of blood are lost. Dysmenorrhea is a clinical condition by which a patient has painful menstruation, and it's a clinical diagnosis that's indicating something non-specifically. There can be a lot of different causes that can cause difficult menstruation, or dysmenorrhea. The proliferative phase is when something is being built up. So the epithelial cells of the uterine glands are now going to multiply, and they spread across the endometrial surface. This serves to restore the integrity of the uterine epithelium after it's been shed during menses. During this time, there's additional growth and vascularization, and this completely restores the functional layer of the endometrium. This also occurs simultaneous to the enlargement of the tertiary ovarian follicles. During the proliferative phase, we have stimulation by estrogens that are secreted by the ovarian follicles. And so the entire functional layer of the endometrium is highly vascularized, so those small spiral arteries are going to spiral in towards the inner surface, and they come from the larger arteries in the myometrium. During the secretory phase, the uterine glands will enlarge, and this increases the rate of secretion. The arteries of the uterine wall will also elongate, and they spiral through the functional layer. So this begins at the time of ovulation, and it extends out as long as that corpus luteum remains intact. So secretion is usually going to peak about 12 days after ovulation and will last for about 14 days, or up until the point where a pregnancy occurs. So this ends as the corpus luteum will stop producing hormones. Figure 2823 kind of wraps up for you the menstrual cycle. So what we're looking at here, starting at the left, is a myometrium in the menstrual phase. So the first day of a menstrual period is day 1. So at this point, what you see here in the histologic image is that the functional layer of the endometrium is pretty much shed, and all we have here primarily is the basal layer of the endometrium. When we move over into the proliferative phase, we have the endometrium and the functional layer having a lot of active glands. And then we have the secretory phase, which will extend about 12 days after ovulation. And at this point, we have all of this accelerated rate of secretion in the glands, which are enlarging in preparation for either menstruation or fertilization. The female reproductive cycle is controlled by hormones from both the pituitary gland as well as the gonads. And together, this forms a complex pattern that's coordinating the ovarian and uterine cycles. Gonadotropin-releasing hormone from the hypothalamus will have changes in its pulse frequency as well as in its amplitude over the course of the ovarian cycle. So for example, the presence of estrogens will increase the pulse frequency, whereas the presence of progesterones will decrease the pulse frequency of gonadotropin-releasing hormone from the hypothalamus. There are groups of endocrine cells in the anterior pituitary, or the adenohypophysis, and these respond to different gonadotropin-releasing hormone pulse frequencies. They'll be sensitive to some, and yet non-responsive to others. Early on in the follicular phase of the ovarian cycle, the estrogen levels are quite low. So the gonadotropin-releasing hormone pulse frequency is between 16 to 24 per day, so that equals out to be about 1 pulse every hour to hour and a half. So as the tertiary ovarian follicles form, a couple things are happening. The concentration of the circulating estrogens rises steeply, and this triggers the gonadotropin-releasing hormone pulse frequency to increase from 16 to 24 per day up to 36 per day. And so this means we're having 1 pulse every half hour to hour. The increased pulse frequency of the gonadotropin-releasing hormone will then stimulate luteinizing hormone secretion. Around day 10 of the ovarian cycle, the effect of estrogen on luteinizing hormone secretion is going to change from inhibition to that of one of stimulation. So this switchover is going to occur when the levels of estrogen go past the threshold value for about 36 consecutive hours. This equals out to be around day 14, which is our average ovulation day, in which there is a massive release of luteinizing hormone from the anterior lobe of the pituitary gland or the adenohypophysis. The surge in luteinizing hormone is going to trigger a few things. The first is that meiosis 1 will be completed by the primary oocyte. The second thing that it triggers is the rupture of the follicular wall, which results in ovulation. So ovulation occurs 34 to 38 hours after luteinizing hormone surge occurs, and this is about 9 hours after the luteinizing hormone peak. Just so you recall this, a personal note for me is when I went through in vitro fertilization to conceive my son, one of the things I had to do was give myself an injection that was called a trigger shot. And what it was a big dose of luteinizing hormone. And so we would time the extraction of ovarian follicles to be about 35 to 36 hours after the luteinizing hormone surge. So that way, it's at peak time for all of those follicles to be extracted. The follicular phase is going to be stimulated by follicle-stimulating hormone, which stimulates follicular development. So monthly, we'll have some tertiary ovarian follicles develop, and as these follicles grow, the thecal endocrine cells will produce androstenedione. And this is an intermediate steroid hormone that's absorbed by the granulosa cells, and it becomes converted to estrogen. So therefore, there are some types of tumors that are present in the ovaries that occur, and these can be called thecal tumors. And if they're thecal tumors, we know that they are primarily composed of tumors of these thecal endocrine cells, and they'll therefore produce a large amount of hormones. Thecal endocrine cells are present in the ovarian stroma, and these thecal endocrine cells will secrete small amounts of estrogen under normal conditions. The circulating estrogens that arise from this are going to be bound primarily to albumins, which is a protein, and lesser amounts are going to be carried by GBG, or gonadal steroid binding globulin. There are 3 types of circulating estrogens: estradiol, estrone, and estriol. Of the circulating estrogens, estradiol is the most abundant, and it has the most profound and pronounced effects on our target tissues. This is also the dominant hormone in our bodies prior to ovulation. Estrogen is synthesized as androstenedione is converted to testosterone, and it does this because aromatase, which is an enzyme, of course, because it ends in ase, converts testosterone to estradiol. So estrone and estriol are synthesized directly from androstenedione. There are 5 primary functions of estrogen. The first is to stimulate bone growth and muscle growth. The second is to maintain secondary sex characteristics, for example, body hair and adipose tissue. It also affects CNS activity, which is especially present in the hypothalamus, where estrogens are there to increase our sexual drive. Also, it maintains the functional accessory reproductive glands and organs, and initiates repair and growth of the endometrium. So keep in mind, as women age and estrogen rates decline, we have a whole constellation of effects, one of which is osteoporosis is quite common in women who are postmenopausal, and muscle mass also deteriorates. Secondly, you'll notice that body hair will decrease, and adipose tissue can change as well. Sex drive may typically decrease when there is less estrogen present. And then last, we'll of course have no endometrial buildup each month because after menopause, this whole process ceases. The luteinizing hormone surge that triggers ovulation also supports progesterone secretion, and then this in turn triggers the development of the corpus luteum. The low frequency of the gonadotropin releasing hormone pulses will stimulate luteinizing hormone secretion, and it does so more than it stimulates follicle stimulating hormone secretion. So therefore, the luteinizing hormone is able to maintain the structure as well as the secretory function of the corpus luteum during this time. During the luteal phase, progesterone levels will remain elevated for about a week. And if a pregnancy occurs, this is different, but presuming that it doesn't, then the corpus luteum will begin to degenerate, and subsequent to that, progesterone and estrogen levels will drop. Gonadotropin releasing hormone pulse frequency will go up at this point, which stimulates follicle secreting hormone, which supports the ovarian cycle to begin anew. Figure 2824, which is a spotlight 2-page spread, really pulls together the hormonal regulation of female reproduction. So first, let's look at the top. We've got gonadotropic hormone levels, and that includes FSH and LH. And what I'd like you to note here is that you'll see that right at the time of ovulation, which is shown by a dotted line running vertically through the image, you see that the luteinizing hormone surges just before that point. Also, we have a surge in FSH that coincides with that. However, we have gonadotropin releasing hormone pulse frequency rise up into just before ovulation, and then it decreases back down again. The follicle stages that coincide at this point is you can see that the tertiary ovarian follicle develops. It finally ovulates around day 14, and then the corpus luteum develops because it's preparing for a pregnancy. And then you finally have a mature corpus luteum just shortly after day 21. And then, in the absence of a conception and embedding in the endometrium, then that corpus luteum degenerates into a corpus albicans. And then down at the bottom, we have our ovarian hormone levels. So you can see that estrogens rise considerably all the way through the very first day of the cycle up until just before ovulation. And then inhibin also rises considerably just up until the point of ovulation, but has a secondary spike just shortly after day 21. And then progesterone, and this is the totally opposite hormone here to everything else. You'll see that progesterone is rather low, but it increases and increases and increases after about day, let's say 13 or so. And this is because we have a corpus luteum that is developing and secreting progesterone. And it's worth noting that that progesterone, which drops back down after about day 22, day 23, that would be about the time when you would begin with a very early pregnancy test to be able to detect human chorionic gonadotropin, which is a hormone produced by the placenta. So at this point, since the body recognizes that a pregnancy has not occurred, it's dropping back down. However, in the event of a pregnancy, then the progesterone level would remain elevated and in fact go up considerably more during the pregnancy. As we just looked at in the previous image, we know that the progesterone levels and estrogen levels will decrease as the corpus luteum degenerates, because remember, it was that big yellow structure in the ovary that is full of cholesterol, which synthesizes hormones. So as that corpus luteum degenerates, those hormone levels go down, and this results in menstruation. So the endometrial tissue shed will last for several days, and that ranges from person to person as well. And it'll continue to shed until the levels of the estrogens continue to rise, and those rising levels finally hit a point at which it stimulates regeneration of the functional layer of the endometrium, and then we restart. The proliferative phase will continue until the point at which progesterone starts the secretory phase. So this increase in estrogen and progesterone will cause an enlargement of the uterine glands, and it also causes an increase in the secretory activities. Over the course of the 28-day cycle, there are fluctuations in core body temperature. So during the luteal phase, progesterone will dominate the entire scene, but however, during the follicular phase, estrogens dominate, and this results in a basal body temperature decrease of about 0.3 degrees Celsius, which is quite minor. Now, upon ovulation, the basal body temperature will have a very quick decrease, just a very quick drop. And a day after ovulation, the temperatures will rise and they remain high through the luteal phase through the end of it. So let's look at the lower component of figure 28-24 so we can evaluate how the endometrial changes correlate with basal body temperature. So what we see up at the top here is at the very beginning in the menstrual phase, you can see the functional zone of the endometrium is destroyed. And as this happens, basal body temperature is just hanging out for the most part. And then we enter the proliferative phase of the endometrium. And you can see that this is when we have repair and regeneration of the functional zone, and again, the basal layer is unaffected. And so it's building up in preparation for potential conception. At day 14, you'll notice that the basal body temperature, you'll see that little drop. And this is right at the point of ovulation. And then after that, it goes up and remains high. So it's expected to remain high for a number of days up until the point at which the body recognizes that no pregnancy is present, at which point it will drop back down. And then once it goes back to normal, or baseline, you can say that it is an indicator of an absence of pregnancy. However, for people who chart their basal body temperature while trying to conceive, they will note that that basal body temperature will remain elevated consistently if a pregnancy occurs. Now let's look at section 28.6, the physiology of sexual intercourse. The autonomic nervous system controls both the male and the female reproductive systems, and sexual intercourse is defined as when semen is introduced into the female reproductive tract. So male sexual function will be coordinated by complex neural circuitry and reflexes, which includes both the sympathetic as well as the parasympathetic divisions of the autonomic nervous system. Male sexual arousal will lead to a parasympathetic outflow over the pelvic nerves, which leads to increased blood flow to the area, and that leads to erection. Male sexual stimulation will initiate the secretion from the bulbourethral glands, which lubricates the spongy urethra inside the penis, as well as the surface of the glands, and this leads to emission and ejaculation. Emission is separate from ejaculation. So emission occurs under sympathetic stimulation, and peristaltic contractions of the ampulla from the ductus deferentia, or the vas deferens, will push the seminal fluid as well as the sperm into the prostatic urethra. So contractions in both the seminal glands and the prostate will pump the seminal mixture along the urethra, and sympathetic contraction of the urinary bladder as well as the internal urinary sphincter is what prevents the passage of semen into the bladder, so it doesn't go retrograde. After arousal, the ischiocavernosus and bulbospongiosus muscles will contract in a rhythmic fashion to cause an ejaculation. This both stiffens the penis and presses the semen towards the external urethral opening. Orgasm is a pleasurable sensation that resolves afterwards in the resolution phase. And this is when the heart rate and blood pressure will decrease and return to normal. Additional blood flow will leave the erectile penis tissue, and detumescence is the formal term for the process of losing an erection when the blood flow leaves the erectile tissue. You may have noticed a lot of commercials on television lately advertising medications for erectile dysfunction, or ED. This has also been called impotence, which is defined as an inability to either achieve or to maintain an erection. This can be caused by a broad variety of physical or psychological factors, so any problem with erection should be something that you visit your physician for to follow up on. Because while it could be something psychological, it's also altogether possible that you could have a vascular condition that you can identify early on through the symptom of erectile dysfunction, and that could save your life. So please do visit your physician if you need to for something like this, and don't be shy. In female sexual function, parasympathetic activation will lead to engorgement of the erectile tissues, just like as in male sexual function, and increased secretion of cervical mucus glands and the greater vestibular glands. In female sexual function, the vaginal walls will have their blood vessels fill with blood, and fluid will move from the connective tissues to the vaginal surfaces, and this provides lubrication. Similar to the male orgasm, the female orgasm is accompanied by peristaltic contractions, and in this case, the uterus and the vaginal walls are contracting, and rhythmic contractions of the bulbal spongiosus and the ischiocavernosus muscles also participate in the female orgasm. Contraception is really just any method that prevents pregnancy. So this can range from abstinence, which is avoiding sexual intercourse, barrier methods, which place a barrier physically in between the male and female gametes, intrauterine devices, which create an inhospitable environment for a conceptus to implant, chemical methods, such as the oral contraceptive pill, and surgical methods, which can include things like vasectomy or tubal ligation. Infertility is defined as the inability to conceive or to be able to carry a pregnancy to term after 1 year of unprotected intercourse. And it affects a lot of people, in fact, up to 10% of the US population, and it occurs in both males and females in a variety of different ways. So, I have already mentioned to you several times how I went through in vitro fertilization to conceive my son. I am more than happy to speak with any of you or all of you at length. Any questions you have, I just think it's something that you should know about because, quite honestly, some nursing programs will not include information about infertility treatments and fertility measures aside from things like the rhythm method. So please do speak with me if you're interested. Consider it an open offer. Amenorrhea is defined as the absence of menstruation, and primary amenorrhea is when menstruation never occurs in the first place. Transient secondary amenorrhea is exactly what it sounds like. It's transient, it's passing, and it's secondary to something else. So this is an interruption of 6 months or more of a normal cycle, and it can be caused by a number of different factors, including weight loss and emotional stress. And when I say weight loss, what we're talking about here is things like bulimia or anorexia, and severe emotional stress or things that would cause PTSD are what we're talking about. Studying for an exam typically isn't going to interrupt your menstrual cycle, but severe emotional stress on an ongoing basis, such as being in a war zone, for example, these sorts of things can absolutely lead to transient secondary amenorrhea. Sexually transmitted diseases are also referred to as STDs or STIs, which are sexually transmitted infections. And these infections are transferred between individuals by sexual behaviors. And there are at least 24 different types, and they include bacterial infections, viral infections, and fungal infections. One example is chlamydia, which is a bacterium, and what it can do is it can cause pelvic inflammatory disease, which basically causes an infection in the uterine tube, which can result in infertility. Let's look at section 28.7, development and aging. In Chapter 28, we're going to limit our discussion to changes in reproductive function, such as the development of genitalia. We'll discuss development and inheritance in more detail in the next chapter, which is Chapter 29. Go ahead and read ahead. It's really fascinating, and I think you're going to love it. So, the development of genitalia. So first off, it's important to note that up until about, let's say, 6 weeks of development, embryos are totally sexually indifferent. So this means that when you look at them through imaging, you cannot identify at all whether it's a male or female. The gonads and other internal structures as well as external structures are both totally similar, and it is the genetic makeup which will determine whether a male or female set of gonads develops. Embryonic testes will begin secreting testosterone, and therefore a male reproductive system develops. However, in absence of testosterone, female structures will form. So keep this in mind as you study pathophysiology in your subsequent semesters, specifically in nursing, where you learn about a broad variety of developmental conditions which may have arisen because of the absence of testosterone. Figure 2826 shows you a breakdown of how at 6 weeks we have a cloacal opening, similar to birds for the most part, that gets subdivided into a posterior anal membrane and an anterior urogenital membrane. So then things will differentiate after 6 weeks, and we develop male genitalia in the presence of testosterone, and we develop female genitalia in absence of testosterone. So you can look at this image, and the takeaway I'd like you to focus on is down at the bottom, the table that compares male and female genitalia. So you can see here how they develop similarly from the same structures, and we end up with different tissues in the male and female bodies, but they're ultimately from the same origins. So for example, the corpora cavernosa is comparable to the erectile tissue of the clitoris for the female. And the corpus spongiosum is comparable to the bulbs of the vestibule. The spongy urethra is comparable to the vestibule, and on it goes. So take a moment to take a look at this just to see the similarity. I think you'll find it really interesting. Now let's discuss the descent of testes. So testes originate inside the body cavity near the kidneys. And the gubernaculum testis is a bundle of connective tissue fibers that stretches out from each testis to a pocket of peritoneum, and it holds the testes in place as the fetus grows. However, during the 7th month of gestation, the fetus grows at a rapid rate, and circulating hormones stimulate the contraction of that gubernaculum testis. And therefore, each testis, the right and the left, will move through the abdominal musculature into the scrotum, and this is accompanied by pockets of peritoneal cavity. Now let's discuss the effects of aging on our reproductive function. In the female reproductive system, the changes that are experienced are all associated with menopause, which is the cessation of the monthly menstrual cycle. In the male reproductive system, there are comparable changes that occur, and they occur gradually, but they extend out over a longer period of time. In the male climacteric, or andropause, which is the male equivalent of menopause, is a period of declining reproductive function that occurs in between ages 50 and 60. And during this time, circulating testosterone levels will drop, and the circulating FSH and LH levels will increase. During this time, sperm production continues, and sexual activity, or at least interest in it, will gradually decrease, and this is associated with declining testosterone levels. Perimenopause is the interval that immediately precedes menopause, during which the uterine cycles, as well as the ovarian cycles, become really irregular. This is because of a shortage of primordial ovarian follicles, and this is something that can be measured by a simple blood test for measuring anti-Müllerian hormone. During this time, the levels of estrogens will decrease, and ovarian may not even be triggered at all. Menopause is defined as a time at which ovulation and menstruation cease, and is usually clinically designated by 1 full year of no menstruation. This is typically present by ages 45 to 55, and by 50 there are really no primordial ovarian follicles left. However, in premature menopause, which was my personal reason for seeking in vitro fertilization, depletion can occur before age 40. At this point, we have circulating concentrations of estrogens and progesterones that are significantly declined. However, the production of gonadotropin-releasing hormone, follicle-stimulating hormone, and luteinizing hormone rise sharply. This is analogous to stepping your foot on the gas and basically not getting anywhere. Menopause creates a decrease in the level of estrogen, which has far-reaching implications throughout the body. It reduces the size of the uterus, as well as the size of mammary tissue. It also causes a thinning of the urethral and vaginal epithelia, and decreased levels of estrogen reduce the rate of bone deposition. So this is why we see commonly women who are older or postmenopausal having osteoporosis at rates considerably higher than younger women who are premenopausal. Lastly, let's look at intersystem interactions. In normal human reproduction, it requires that we have males who have a sperm count that's adequate, the semen has to have the correct pH and nutrients to support the sperm, and erection and ejaculation both must function properly to be able to deposit semen. In females, we expect that ovarian and uterine cycles have to coordinate properly, and that ovulation and oocyte transport have to also occur normally. The environment of the reproductive tract also has to be able to support two things: the survival and the movement of sperm, as well as the fertilization of an oocyte. So I think we can suffice it to say that in human reproduction, it's complicated. There are a lot of systems at play. The reproductive system, most notably, but also the digestive system, the endocrine system, of course, which runs the reproductive system, the nervous system, which also runs the reproductive system, cardiovascular system, and the urinary systems. But even when all systems are go and fertilization occurs, a healthy infant is only going to be produced if the zygote is able to develop into a full-term fetus. We'll discuss more about this in the following chapter. Thank you so much for your attention, everybody. I really hope you enjoyed this lecture. I know there's a lot of content this week, but I'm hoping that you knew a fair amount about this topic ahead of time, so I'm just kind of backfilling in the gaps in between your knowledge. This photo here, by the way, is of me with my son Ezra very shortly after he was born, and I brought him home. And I call this picture From IVF to Ezra, because what we have here is the two blastocysts, which were implanted in me during IVF, and one of those became Ezra. I don't know which one, so take your guess. Whichever one looks better, I guess. But anyway, if you have any questions, please give me an email. I'm happy to talk with you or Zoom with you anytime. Have a wonderful week, everyone.